Good morning, everyone. So let's get started. Okay. So yeah. So I mean, I know this is a bit early for the West Coast people. So really appreciate. So you come here on time. So let's get started. So before we start this today's session, right, it's another exciting day. And I think Masala and I myself are going to do a little recap of what we had yesterday. And, just to refresh our memory, right? So as a refresher. So, and we have about 10 minutes. So I will, maybe I can start a little bit and then Masala. So the first session, actually, Carl Young and I myself co-chair the first session on recent advances in AI and application to biomedicine. So we have 4 talks. Actually, if you look at the screen, those are the 4 talks, 4 wonderful talks we had. So, after listen to these 4 talks, we just have some, a recap of some take-home message, right? There's many things we have discussed, a very wonderful discussion with the panel. So at least this is personally what I learned. Important, point would be like, it appears that a large language model or in general foundation model, right, has taken over, right, to, to really demonstrate its power, right? So not only from like the all the way from the keynote speech and also the presentation by Dr. Zhiyong Lu, right? So they demonstrate actually already successful initial result of using large language model to help to address some of the fundamental questions in the biomedical field, or facilitate search and query and assembly of knowledge, and using that to make a prediction and inference, right? So that's going to continue to be the trend, right? So to see the power of large language model, the foundation model. The second piece of information I learned was be like actually the heterogeneity in data, right? So different data modality, right? Used to be in the classical machine learning field, right? So we focused on single modality, but now actually the power of harnessing multiple data modalities is going to be the trend for the future. Of course, this is going to help us, if we do it right, to address the heterogeneity, not only in the data, but also in the disease that we really care about, right? And the next point is that, actually this I learned from Professor Jie Liu's talk, is that actually extracting structured knowledge from unstructured data is critical. It's going to bring additional benefit than, you just use your text data by itself, right? So he shared with us some of his exciting initial results, as well as I saw in the poster session, there are some more of the details being shared. So I think that's also an exciting direction to follow. And last but not least is the ethics in AI, right? So it's how do we address data and model bias? How do we promote equity, right? So that's also critically important. And the last, we also, discussed by Professor Hongfang Liu about the practical considerations of deploying AI system into our healthcare system. So I was, this is my quick summary. So I'm going to hand over to Masara.
Thank you, Wei. First of all, I find this workshop very interesting and very timely because it brought together people across different disciplines. And it's timely because just 2 Nobel Prizes were awarded to scientists who involved AI in their research. 1 Nobel Prize went for physics, and the other one went for AlphaFold for chemistry. And I'm very proud that a graduate of Vanderbilt University is the youngest scientist awarded the Nobel Prize in past 70 years. So this is remarkable that this workshop came together so timely. And in terms of yesterday's discussions, I thought it was really fascinating session because we were able to, let's see if I can advance the slides. Okay, we were able to cover different areas or different forms of diabetes through prism of primarily researchers, scientists who are involved in the clinical and translational research. So here are the forms of diabetes that we think about and that I, as an IELTS biologist, think about all the time. But we do have significant challenges to navigate this field. And these challenges come through us, considering these different forms of diabetes, how they're diagnosed. Currently, only glucose is really a diagnostic tool. And we do not understand fully. We know beta cell plays central role in all forms of diabetes, but what we do not currently understand are molecular signatures and genetic features in diabetes heterogeneity. So these are really true challenges for us. And just thinking about yesterday's session, how can we face these challenges and seek new opportunities? I think true opportunity for us is really for the first time to connect genetics through genomics to cellular function, to understand underlying mechanisms of personalized signatures for diabetes predisposition. And how do we bridge really basic science and clinical and translational research? And I think AI is going to be great in this realm, right? Because on the side of basic science, NIH invested into remarkable programs such as Human IELTS Research Network. There is Integrated IELTS Distribution Program. There is Human Pancreas Analysis Program, a HubMap that can really provide a lot of valuable data about IELTS function, morphology, physiology, and genetics. This is all dependent on organ donation in this in the United States that had been really orchestrated through organ procurement organizations here. And then on clinical side, we have programs like Radiant, we have TEDdy, TrialNet, Inodia in Europe, and biobanks around the world, including UK Biobank. We have FinGen, BioView, and the Hong Kong Biobank that we heard about yesterday. So how do we do all this, right? It will require building interdisciplinary teams and collaboration across interdisciplinary teams. And how can AI bridge the gap? Well, I think through biomarker discovery, right? Through development of diagnostic tools that will allow us to diagnose individuals with diabetes much sooner and develop therapeutics that would protect beta cell function. And I think our end goal should be really prevention of diabetes, right? So those are my 5 cents for yesterday's sessions. I thought it was a great day yesterday, and seeing collaboration and bringing scientists from different disciplines together. I think that's the path forward, right? All right, thank you.
All right. Now it's my pleasure and honor to introduce Dr. Greg Germino, Deputy Director of the National Institute of Diabetes and Digestive and Kidney Diseases, who will deliver opening remarks today. I also want to mention Dr. Germino also oversees the data science working group of the NIDDK. Greg, thank you.
Good morning, and I'm just going to be just a few moments. I just wanted to, again, welcome everybody who's here, and thank you for participating. Those of you online, thank you for participating. As you know, this is an important concept, and it's something that we at NIDDK all the way through the organization see as important for our future. You know, as we know, one of the greatest challenges in modern medicine is the complexity of disease. We've been talking about diabetes mostly yesterday, and we're going to be talking about it some more today. But in fact, and we know that's not a single disease, but that's true for many, many conditions that we have in NIDDK, whether they're talking about obesity and weight control, whether we're talking about chronic kidney disease. I'm a nephrologist, so this is something that's, of course, relevant to me. The approach that you're discussing today is one that's generally applicable to many, many things within our condition, which is why from Dr. Rogers through me coordinating our data science working group, this is something that we are prioritizing across all of NIDDK. As you know, this meeting was organized in response to the rapid advancements in artificial intelligence which hold tremendous promise for transforming precision medicine, as we heard yesterday, and particularly for chronic conditions like diabetes and chronic kidney disease. The goal of this workshop is to bring together biomedical researchers and AI/ML experts to discuss the critical challenges, key gaps, and opportunities for using AI to improve disease prediction and treatment. Our focus is on how AI can help us better understand the intricate preclinical pathways that contribute to disease development, uncover disease subtypes, as we heard yesterday, and identify early biomarkers, which are important not only for identifying patients and individuals most at risk for progression, but even for the design of our clinical studies in terms of interventions that we want to use as endpoints or measurements. You know, as you heard from Zhijing, as the deputy, I've been managing our data science efforts over the last, more than half a dozen years. In fact, my first sort of opportunity to think about this was with a former program officer in DEM, Ron Margolis, who dragged me to one of the dkNET meetings, I don't know, a dozen years ago after I'd gotten here and said, listen, Greg, you got to think about this. This is really something that's important for all of NIDDK, this data science space. And of course, that's grown tremendously. And we have, over the last half a dozen years or so, had an internal working group. It's been marked by having a data science forum with our advisory council, again, just raising the awareness and highlighting the importance of these technologies and these approaches to science to the key parts of our organization. And in fact, we have an effort underway right now at NIDDK to understand what we're doing in the data science space and what we're doing in terms of how do we go forward in this. And of course, AI/ML is at the cutting edge of these technologies. Building all this science, getting all the science, all the data into a standardized way so we can do the new discoveries. Today, you're also going to be hearing about some of how we might leverage what we're doing with other resources that are sponsored by NIH and NIDDK. Dr. Rodriguez, who's sitting in the back, is going to talk about our DK repository, the data repositories that we've had going back 20 years, and importantly, linked biospecimens that can be used for testing, as we just heard, looking for biomarkers and the like. So again, looking forward to the further discussions as we go through today. Finally, I just want to, again, thank all of you for coming. I want to thank the meeting organizers, all the speakers, the panelists. Again, this is a really important and cutting edge technology that I think is going to have tremendous impact. It's going to be a before AI and an after AI, and I think we're just at the cusp of seeing that change. So thank you very much, and again, looking forward to the rest of the day. Thanks.
Right, my pleasure to introduce our session 3 today. Ashley and I are going to be moderating it for you. The first speaker is Cecilia Lee from the University of Washington, and she's going to speak about opportunities for studying heterogeneity, uncovering new biomarkers, and predicting progression in types of diabetes using AI-READI dataset. So, Cecilia.
Good morning. Okay. So I want to thank the organizing committee members for this opportunity. So Bridge to AI program is an NIH Common Fund initiative that recognized that the potential of using AI and ML in healthcare or biomedical research, but also recognized the lack of data sets that are AI ready and then really ready to target complex medical problems such as diabetes. So Bridge to AI program has 2 components, Bridge Center. I put the pictures of Chen and the Pei Ping because they are part of Bridge Center, and then 4 data generation projects. So I'm here to discuss AI-READI project, which is one of the data generation project. So I'm gonna go with our data generation project. Our data generation project is called Salutogenesis Grand Challenge. So our goal is to create multidimensional, ethically sourced data set in diverse people for studying salutogenesis in type 2 diabetes. We call ourselves AI-READI as AI-READI and Equitable Atlas for Diabetes Insight project. So we are very familiar with the concept of pathogenesis as researchers in healthcare. And we are familiar focusing on risk factors, and salutogenesis is the opposite. You can think of, what are some factors that would promote health from disease state. So the concept behind or the design behind our study is borrowing the success of single-cell RNA-seq field. So when you are able to characterize all these cells and you use the pseudotime manifolds techniques in machine learning, then you can put all the cells that are very similar to each other, very close to each other, and if the cells that are different from each other very far. And then we can create the pseudotime manifolds atlas or pseudotime atlas of the cell progression from embryonic stage to totally differentiated stage. So we can kind of imagine how we could do that if we have large enough sample and characterize them to exquisite details. And we could create the pseudotime atlas of a person going from health to disease and then vice versa. So our concept is that we are going to create multimodal data that is AI ready or machine ready. And you can use those data sets for cross-sectional AI models for hypothesis generation and biomarker discovery, but also using pseudotime manifolds technique, you could do predictive AI models in terms of who are going to develop progression, and then how do you predict somebody's outcome. So I'm going to give you more details of our study. So we were very focused on current limitations of currently existing large data sets. So we are balancing our data set very carefully. So our goal of recruitment is 4,000 participants, equally balanced in race and ethnicity. So 1,000 white, 1,000 Black, 1,000 Hispanic, and 1,000 Asians, and also equally balanced in men and women. And importantly, we are balancing across the disease states. So 25% non-diabetic, 25% pre-diabetic, and 25% medication controlled, and 25% insulin controlled. We're going from head to toe. So I'm going to give you this spectrum of how extensive our data collection is. Once before visit, they complete a lot of surveys, including the depression scale scores, diabetes score, and a lot of social determinants of health. When they come, we do vitals, waist to hip circumference. We do ECG 12-lead EKG, MoCA, and monofilament tests, and visual function, and lots of retinal imaging. And we obtain blood tests and urine, and then obtain things like C-peptide, CRP, hemoglobin A1C. And we are also banking blood, Buffy code, plasma, serum, and PBMCs for future iPSC work. And they go home with 3 devices, one Dexcom continuous glucose monitoring device, and physical activity monitoring that they will wear at the same time of CGM. And then also our in-home created device for environmental sensor. I'm going to touch upon that a little bit later. So I used to be very excited about this slide because this data set of the pilot data over 200 people just got released this summer. It has over 500,000 glucose measurements, over 33 million air pollution data, and it was about 311 gigabytes of data. Since this summer, over 110 research organizations around the globe have downloaded our data set. But why am I not as excited? Because this is coming next month. So our full data collection of year 2 is on 1,067 participants. We have near 3 million glucose measurements, over 42 million respiratory rate measurements, and 182 million air pollution light spectrometer readings. And I'm very biased, obviously, for retinal imaging, but I'm going to say that this is the largest OCT angiography database in the world. And this is 2 terabytes of data that you will be able to download next month. So now I'm going to kind of show you some showcases of the potential of using our data set for studying various aspects of diabetes. And I do want to emphasize that these are all from our QC data, QC work in preparation of data release. We have not published any of these. These are really for you to study. And then we're really trying to move away from this concept of holding our data set until we have published it all and then maybe sharing it later. No, we are sharing every year, every time we do data collection. This is work of Aya Matayoshi from our work. So this is a distribution of our age on our over 1,000 people. This is a BMI, glucose, so hemoglobin A1C. So not quite surprising, right? So there's a nice distribution, but them skewing to the right-hand side. One thing that surprised me was how prevalent depression is in our cohort. So that's another, thing that we could study. I'm glad everybody is very interested in CGM, and we are very interested in as well. So the Dexcom device has glucose reading every 5 minutes for 10 days. So this is work of Julia Owen in our lab. And now this is a histogram of people in no diabetes group, how frequently they were in time in range. So obviously about, the majority are in time in range in people with no diabetes. Pre-diabetes, okay, a little bit less. Medication group, oh, maybe not as good. Insulin group, all over the place. So obviously there is lots of heterogeneity even among the group. And so let's look at time in tight range. Even no diabetic group, there are a lot of people that are not in tight range. So what are the biomarkers? What are other characteristics in these people? Pre-diabetic, medication, and insulin group, they are most frequently not in time in tight range. Another way of just, doing QC of this data is, creating this beautiful scatter plot of all the data points. We ran out of colors because there are too many people. So excuse that. And so this is the 10-day worth of data, no diabetes, pre-diabetes, medication, and insulin. So it's a beautiful though. You can see this progression and insulin just, there's a painting all over the place, right? And one thing that was really surprising to me is that in no diabetes group, do you see that purple line at 400? There are people who are living in 400 range and they claim they don't know that they have diabetes. So things like that. So we are sharing raw data so you can reclassify as you want to. What is your definition of diabetes? We talked about, it's unclear. Is it really blood glucose? Is it other blood measurements? So this is here. What about uncovering new biomarkers? Can we use our data set for this? So we know that environmental factors play a significant role in health outcomes, not just diabetes. But most studies really relied on outdoor conditions, the measurements on OSHA or the, NASA. And then the measurements are very crude and then they are on the zip code level. So we created a home-based sensor for developing more granular data. And this collects data points every 15 seconds on 22 different home pollution and light spectrometer variables, such as PM 1.0, 2.5, 4, and 10, and volatile organic compounds and nitrogen oxide. So this is an example. So it's the size of 2 iPhones. It's very light. Participants love it. They just plug it in and then they just forget about it. But are we getting enough data and interesting data? So this is the work of Jamie Schaefer from our lab. So we are creating this concept of individualized exposome in their home. So I'm showing 2 participants. One participant A have lots of PM 2.5. Participant B really barely nothing. And there is this, definite difference that you can tease out. Is this participant more exposed to nitrogen oxides, VOC? So those could be another biomarkers. So what if you look at the group level? And we are really surprised by this. So let's look at, mean max of particular matter. And, we can go particular PM1 or 2.5 and all of that. And we are seeing difference. So insulin control group seems to have more exposure to PM 2.5. And then if you're looking at volatile gas mean and 75 percentile, also we are seeing signal. So these are things that are surprising, and then you could come up with a lot of hypotheses why, and we could create another, data set to prove that. But these are kind of the richness of data that exists that are really here for you to leverage. Retinal imaging, I told you I'm biased, but retinal imaging really is a unique opportunity to get multimodal information, not only about retinal tissue and then neural tissue, but also vasculature. We are able to look at capillaries and image them in a matter of seconds. And this is an image of OCT, or optical coherence tomography. And then the right side is the histopathology slide of a retina. And you can actually we can actually see those 7 layers on this OCT, all non-invasively. And then, our imaging goes up to 2 to 5 micron of resolution. What about predicting progression? So we are doing a lot of social determinants of health data collection with Phoenix Toolkit. So those could be also integrated in your AI models in predicting progression. Again, we are really going from head to toe. So head, with MoCA, with cognition, and then really toe. We had a hard time convincing our technicians to do to take out the socks and then to do the neurofilament test because they are used to eye data, but these are going to be really available for all the researchers to study. And we are planning on whole genome sequencing in the year 4, and then also EHR grab. So we're going to do pool of electronic health records from all of them. And also the specimen have been banked. We have over 7,500 biospecimens banked so far. How do you access our data set? So our data set is divided into publicly accessible data set and then restricted access data set. But all the data that I have just told you, that I've shown you, they are publicly accessible and downloadable. I don't have time to go over all the security, all the privacy concerns that we kind of dealt with. I'll be happy to talk to you more, but we really wanted this data set to be AI ready and accessible. I heard from the small group yesterday, CGM data, you can't even get it. It's so hard, it takes years. We are really trying to get rid of that barrier and then really accessible and promote the science. So you can really go and then take a look at our pilot data. It's going to be the same format as our year 2 release. So if you want to play around, create the code, it's going to be ready to go when the year 2 goes. So, and then we did a lot of standards and harmonization work so that it could be AI ready as well. So I want to acknowledge all the team members and, Mike Snyder, who's here. He's also part of our consortium. I really want to thank for this opportunity, and I hope everyone is excited, and then hopefully there will be a lot of opportunities for you to leverage this data set. Thank you so much.
That was excellent, Cecilia. I want to remind everyone before we go into the next speaker that this is a panel discussion, so you get a chance to ask questions at the end, and we'll bring them up like we did yesterday. So because this is a session spanning data science programs across the NIH and NIDDK, we'll talk about how to pull them all together and impressions on all of them at the end. Our next speaker, though, is Dr. Chen Li. He's going to talk about dkNET and its scalable cloud-based computing platform to support the integration of AI, ML towards accelerating discovery and translation.
So thank you for having me here. So I'm Chen Li from UC Irvine. My background is more about data systems. I'm a data guy, and we build software, we build infrastructures to support data science and data management. So I noticed my title is a little bit too long, so I gave a shorter version of the title, which is our dkNET portal and is newly introduced computational core to support collaborative data science and AI/ML. So the overview of my talk is first I give a introduction of the dkNET portal, and I'm going to introduce this new computational core we are actively working on, and I'm going to explain some of the key features and capabilities of this new core related to data science and AI/ML, and I'm going to use a particular use case to explain its power. dkNET, for people who don't know what dkNET is, it is a portal we started mainly from San Diego more than 10 years ago. dkNET provides this portal with all kinds of information related to data, materials, funding opportunities, news, and projects. It provides tools and services to support in support of rigor and reproducibility built around the research resource identifier, or RRID, and the FAIR data principles. And we have a website, dkNET.org, mainly maintained by our colleagues from at UC San Diego. So you're welcome to visit the website to get all the information about this portal. And the activities have been ongoing for more than 1 decade. So in the new phase of the renewed dkNET portal, we are adding a new, very important component called the computational core. So in the next few minutes, I'm going to use one hypothetical situation where we have a PI called Alice, who is a biologist, and she manages a team with people with different backgrounds, including biologists and bioinformaticians. They do all kinds of analysis related to data, in particular data sequence analysis. So we're going to use this example to illustrate some of their pain points and the value this new computational core can bring to the community. So this is one of the example pipelines people in this space are working on almost on a daily basis. For people who are not familiar with this pipeline, at a high level, we are doing this single-cell RNA sequence analysis. So this whole analysis can be viewed as a pipeline of multiple steps. And the first step is doing some alignment from short reads against a long reference sequence using packages like a Cell Ranger or Star Solo. And the data goes through multiple steps, including quality control, normalization, dimensionality reduction, integration, clustering, annotation, and then before we do more advanced analysis using latest AI/ML techniques. You can see the pipeline can go on with the multiple steps. I didn't include some additional steps here. So what are the pain points? There are quite a few. The first pain point is coding. Not everybody in this room knows coding. Not everybody enjoys coding. I'm from CS, and that's even for myself, I found it's time consuming to do coding mainly because of my tight schedule. And coding requires skills in programming languages like R, Python, Java, Scala, Stata, or MATLAB. But many of the people in this space, for all the reasons, either don't know coding or don't have the time to do coding. But unfortunately, there are many latest techniques, especially in the space of AI/ML. Some of the techniques are packaged in some libraries in the Python R. As a consequence, there is a barrier between these domain scientists and the latest AI data science techniques. The second challenge, a pain point, is lack of computing resources. Take Alice lab as example. They may want to do some latest AI analysis, and they could use some of their local computer clusters, but not everybody has the deep pocket to buy a cluster of machines. Not everybody could afford some IT staff people to have the skills to set up and maintain such a cluster. In some cases, they may just rely on the infrastructure provided by their university, shared with some other researchers. But in that case, sometimes they have to wait for the availability of the cluster, or possibly even wait for the availability of the staff members. As a consequence, their work is blocked. The human time can be wasted. The third pain point is very often we have a lot of places where we can share data, but we see the increasing need for people not to just share data, but also share their analysis. In particular, you can see all the final very beautiful charts were produced. They have gone through a very complicated pipeline written, say, in Python or R. And typically what people do is, let's say a senior student, they write, say, Python program or R program, and they're going to graduate. That's our nature of a business, but they're going to put a source code somewhere on GitHub. And nicely, they can write a README file. But the new people who come to the team, they have to go to GitHub and look at the README file. If they have the patience to follow the steps in the README file, and they might be able to reproduce what the senior person has done. But in many cases, as we all know, the new people, they just say, forget about it, let me start from scratch. This situation also happens not just within the lab, also between different labs within the whole community. As you can imagine, a lot of resources, a lot of knowledge that have been invented by our other people, our colleagues, have been wasted. So the question is, how can we provide such an infrastructure to allow sharing, not just data, but also about the pipelines? And GitHub, these kind of infrastructures, they don't provide all the capabilities we want. Given all these observations, we want to bring in this new computational core to our dkNET portal. At a high level, in this new computational core, we want to allow researchers in the community to be able to share their data analysis and also their machine learning models. We also want to bring compute to this platform. We want people to do computation on the platform. And in addition, we're using our homegrown system called the dk-CR to have a workflow interface to lower the barrier for people to conduct the data science and AI ML analysis. And we're actively working on this component to also collect all the data from different modalities in order to develop foundation models and make the foundation model available to the community. And correspondingly, we want to develop some advanced features that can be powered by this foundation model. For example, like a ChatGPT-like interface for people to ask questions related to this community, not just the general ChatGPT interface of OpenAI. So next, I'm going to show you a few very important features this computation core can bring to the community powered by this dk-CR system. So this is a screenshot of a workflow of the one I showed you earlier. I just rotated the layout by 90 degrees. So this is exactly the same workflow we saw earlier to do single cell RNA sequence analysis. And you can see this is a very intuitive interface with multiple steps. Each step is called operator. You can see some of the steps has an R in it because internally that part is written in R. The whole platform supports different languages, including R, including Python, including Java, Scala. These are the commonly used program languages. So in this way, if there's some advanced functionalities you cannot implement using the current existing operators, you have the freedom. Some people can use their own skills of, say, R programming to add some additional operators. So this is a screenshot of one of the real workflows we worked on. So I want you to pay attention to some of the details of this screenshot. So you can see currently there are two people on, I don't have the laser pointer here. On the top right of the screen, there are two people who are actively working on this workflow. This is very similar to the experience other services like Google Docs or Overleaf, if you know what it is, what they can provide, which is we allow multiple people to concurrently work on the same data science pipeline. And also they can obviously they can work remotely. And then on this workflow, some of the operators, if they are green, that means the execution has finished. And also you can see at the bottom of the screen, we have this cursor. The case is I'm seeing what my collaborators working on, similar to the case where on Google Docs, you can see the cursors of other collaborators. And in the middle part, we see some of the operators, they are coming from the popular sklearn package. So we implemented some of the existing functionalities of like sklearn, some other libraries. And those existing AI ML techniques have been prepackaged as operators. From in the user perspective, they can easily drag and drop some of the operators to do the latest, to use the latest AI ML techniques to analyze the data. So on the top right, you can see the share button. As you can imagine, this platform allows you to share this workflow with your collaborators. And we can also publish the data set. Oh, thank you. Thank you. Thank you for the trick. So I need to know where to point to. Thank you. Yeah, I think you get the idea. And on the left, we can see we allow you to manage workflows, data sets, and in the future, we also plan to add ML models. So now I'm going to use a particular use case to explain some additional capability of this platform. The sequence alignment. And for people who don't know what it is, at a conceptual level, it is about how to map a large collection of short reads against a long reference sequence. Okay, there's a one-sentence summary. This process is known to be very time-consuming. In one of our experiments, we noticed that if you use some of the packages like a cell ranger as example, in some setting, it can take hours, days, or even weeks to finish it. Now, you can use one local machine that becomes a bottleneck. You could add more machines. Now, we noticed that we saw the pain point I mentioned earlier. Not every team has the capability to do so. So on the Taxera platform, we have implemented this a new functionality called a Cloud Mapper. From name, you can tell this Cloud Mapper allows people to do mapping using the cloud. So this is a diagram to show how it works. From the end user perspective, they can just come to the site through their browser, and using the Cloud Mapper, they can elastically request more resources from public clouds. In this case, we're using AWS, and they can specify the configuration of each machine. I have a screenshot here. So on the left, you can see what kind of machine configuration they want to choose. And on the right, they can choose how many machines they want to use. In this way, they can just, by doing a few clicks, they can easily start a cluster with that hardware configuration, with a number of VMs. And the cluster will become available for this researcher in just a few minutes. So then they can just deploy their alignment task to that newly formed cluster and finish the job. So we did one experiment. In that experiment, if we use the user's own server, it took more than 4 hours. By using the Cloud Mapper feature of the Taxera platform, it took less than 18 minutes using 16 VMs, virtual machines. The total cost is less than $4. I think these days, given the price we have here, roughly it's less than half the price of a burger. So the beauty is you can do everything elastically, pay as you go. So to summarize, the new Taxera platform for the computation core has currently 3 main values. First, it allows low or no coding for data science, AI, ML, for people with different backgrounds. And second, it has very powerful support of collaboration and sharing between each lab or within the community. And third, it allows elastic computing by grabbing more resources from the cloud as requested by the end users. The current status is we are actively launching this service. We are inviting initial alpha users. So if you have the pain point, you see the potential value of our system to you, please talk to us. And we are also developing more ML models. The recent pilot project we're announcing is related to that space. And we are also working on how to develop and publish foundation models in this space. So again, this is thanks to NIH, NIDDK, and also NSF, and thanks to my collaborators, Jeff, Wei, and Shuibing. Thank you very much.
Thank you, Dr. Lee. Our third speaker is a friend of mine for many 15 years, so I get to add that to the introduction. Dr. Kyle Gaulton from UCSD. He's going to speak about PanKbase and knowledge base to support type 1 diabetes research. Okay.
Okay, thanks, Mo. And thanks everybody for a great meeting. And yeah, it's really fantastic to be here. So yeah, so talk today about a new initiative that Noelle just mentioned, which is called PanKbase, which just started this past year. The goal of which is to build a platform to provide knowledge of the pancreas and hopefully facilitate research in type 1 diabetes. So why is this needed? So there are many gaps in our understanding of type 1 diabetes, pathogenesis, and treatment in the pancreas. So just some gaps that exist currently are understanding mechanisms of disease. So, we know a lot about how type 1 diabetes develops, but there's still major gaps in understanding the specific triggers of autoimmunity and disease initiation and so forth. There are therapies that can delay the onset of disease, but no therapies currently that can prevent disease altogether. There's limited ability at the moment to be able to distinguish, for example, likely progressors to disease, particularly at the very early stages, which is needed for identifying people who may qualify for preventive therapies, as well as understanding heterogeneity. There's differences in disease mechanisms, presentation and treatment, et cetera, based on a variety of factors. So given all these gaps, it's a major challenge to try to fill in this knowledge. There's a wealth of data that's been generated in the human pancreas, both in terms of data repositories that exist. So things like HPAP, PankDB, NPOD, IDP, et cetera, many of which I've heard about over the past couple of days, as well as many different types of data that are collected on pancreatic donors that are made available in these repositories. And so leveraging the wealth of data that these repositories contain and the different types of data that are in them represents a real opportunity to improve our understanding of the pancreas and its role in T1D. So although that's an opportunity, I think there's some key barriers that exist that maybe prevent access to this information and deeper insight in order to further our understanding of T1D. So some of these barriers are the availability of standardized metadata and data in the pancreas across resources. So these data are dispersed across many resources, both pancreas-specific resources as well as more general repositories. And lack standardized processing, quality control, centralization, et cetera. Metadata describing these donors and experiments isn't always aligned across resources, as well as pipelines for processing of these data are not easily available oftentimes. Two, there's the availability of key resources derived from high quality data are often lacking. So there's few what I call integrated resources of high value data that have the standardized processing, including formats to interface with other data and downstream applications, machine learning and AI type tools. There's also often unclear provenance and reporting of data, as well as biases, outliers in data contained in these resources. And finally, access to data. So you need multiple interfaces that provide these data, as well as both basic and advanced representations of knowledge derived from these data. And analyses need to be provided in formats that, and we heard the previous speaker mention this, that accommodate diverse user types, ranging from maybe pre-computed results that are just made available for people to download to maybe workflows that are easier for non-computational scientists to use, to cloud-based workspaces that enable advanced users to be able to run analyses on these data. So you need a variety of mechanisms for people to run analysis. So that is the goal of PanKbase, which again, I said started this past year. The hope is to address some of these challenges. And it is an initiative through HERN. And again, the goal is to develop a platform to provide knowledge of the pancreas to understand type 1 diabetes. It's a multi-institution collaboration, investigators that have a variety of expertise. Actually, many of the people involved in PanKbase are here at this meeting. Expertise in this group spans platform development, type 1 diabetes, data science, as well as people who have expertise in islet biology and diabetes, and also importantly, engagement and outreach. So what are the key areas of focus for PanKbase? So there's a couple tenets I would say that we're focusing on. So first is creating a catalog of highly curated data as well as rich metadata. Two, to develop a platform that a range of different types of users can access this information. So serving biologists, clinicians, data science, et cetera. Developing analytical tools that run in any computing environment that allow people to be able to run computing on these data. Promoting, I would say, standards for human pancreatic research, both in terms of the metadata that's collected as well as the data and how it's processed and QC'd, as well as, again, both engagement in terms of the community. So understanding the needs of people who study type 1 diabetes, who study the pancreas, as well as engagement with experts who have a ton of knowledge that can be tapped into to enhance the platform. So I'm going to talk today about mostly the beginning. So since we just started, which is the cataloging of highly curated data and rich metadata, which we kind of viewed as a starting point for building the rest of the platform on top of. So what do I mean in terms of creating a catalog of resources with high quality data and metadata? So resources could be things representing donor metadata and comprehensive information about donors. So clinical information, demographics, information about biosamples obtained from these donors. Resources could also be things like single cell maps, so like derived products from single cell assays that contain not only the cells, but also annotation of the cell types of these cells and so forth, functional measurements on islets, and many, many more different types of resources that could be created. There's also a variety of external resources that wouldn't necessarily need to be created directly through this platform, but are high value resources. For example, QTLs, for those who are familiar, like genetic associations with say gene expression data or other molecular phenotypes that are products that exist in the community or published that are also valuable to draw in. And I think the key thing here on the bottom is that these resources are drawing data combined across multiple different resources. So in HPAP, there's over 100 donors. IDP has collected at this point over 1,000 donors. Over time, we're incorporating other resources. So Pat McDonald's here. So Islet Core, which is run through the University of Alberta, has over 500 human islet donors and so forth. So creating resources that combine information generated across all these repositories, and then taking these resources and then providing them to users and using that as a blocks basically to facilitate cross-modality analyses that people may be interested in doing. There's also knowledge that can be derived from these resources. So you may have a single cell map, but that on its own may not tell you very much. But you can use these maps to derive things like where's the gene expressed? What's the change in expression of a gene in disease versus control? What's a gene that marks the identity of a cell type relative to others, and so forth. How do we represent these data and knowledge? So there's a couple ways that we are thinking about providing this information to users. I mean, one aspect of this over time is like cloud-based workspaces where people can just access data directly, also download data. There's also a lot of value in visual representations of data and knowledge. And so this is one example developed by Noel and Jason at the Broad Institute for knowledge portals that they developed, where they represent information derived from single cell maps, allow you to interact with them, visualize the information in these maps, subset in various ways. Other ways are through maybe structured representations of knowledge. So Jay talked about this yesterday, building knowledge graphs, which form connections between pieces of knowledge. And PanKgraph is the name of the knowledge graph that's being built as part of this project that Jay's talking about. And so you can use knowledge graphs both to query basic entities, so just like simple relationships, as well as reason over these knowledge graphs. And structured knowledge, which I think was a key thing that was touched on yesterday, to provide accurate representations of knowledge and also provide that information back to users in intuitive ways. Okay, So again, I think a key here is to build the catalog of highly curated metadata and data. And so in some cases, we're creating these resources ourselves. So for example, if you take bulk RNA-seq data or single-cell RNA-seq data, you run standardized processing of these data, generate outputs from these data sets, summarize the data that was collected, as well as applies the uniform quality control, and then create high-value products from these. So in the single-cell space, like, high-quality single-cell maps that have all the single-cell data in it, or even from bulk RNA-seq products that contain all the samples and expression levels of genes in one place for people to use. And if you look on the right here, just in terms of cataloging the number of data sets, I mean, particularly in the single-cell space, I was a little surprised in a good way how many single-cell data sets have been generated just in pancreatic islets. It's well over 300 data sets, and some of that is in diabetes-specific repositories such as ASPAT, but a lot of it is in GEO and data generated from islets that people get through resources such as IDP and also commercial entities. So there's a ton of data across resources that can be used to create these products. I think something that is also quite important, just checking time here, is comprehensive metadata and the recording of metadata that can be tied into these data resources that we're collecting. And so again, this concerns things such as information about human donors, as well as biosamples collected on those donors, in addition to the experiments that are performed on these donors. And in the human donor and biosample space, there are established standards that have been developed and recommended and are actually required for certain diabetes journals for reporting of human islets, and adopting these standards and promoting them and requiring some level of these standards for data that is aggregated by PanKbase, I think will be, again, an important set of building blocks for people utilizing this information to learn things from it. And one thing that I was talking at another symposium the other day while I was putting these slides together, and this slide came up, which I thought was very relevant, in that curating metadata is extremely important, but it's also very tedious and probably pretty boring. But it's also a very important aspect of making sure the provenance of the data is correct, and then also there's a richness of information that it could be used to leverage for analyses. And so we're collecting metadata on all the donors and biosamples, and this is just one example of how we're representing that. Another thing that I think is quite interesting to note is that so many data sets that are published in the literature are not connected to, and oftentimes, the extensive metadata that's been collected on these donors. So a key example of this is IDP, so it's Integrated Islet Distribution Program, which is a resource supported by NIDDK that basically provides islets to researchers who want to then perform experiments on these islets. And then people will go off and do experiments on islets, publish it, like reported in a paper. And oftentimes the identity of the donors that were used in these studies is not tied back to the original donor that they were sent. So connecting data in literature from these donors back to oftentimes the richness of information that was originally collected in these donors is a missed opportunity. And one way that we've been thinking about trying to tie data generated on these donors to the donor that they come from is by using genotype information. So I don't think all the donors in IDP, but many donors in IDP are genotyped. And then if you have genomic data, you can genotype the genomic data, and you can connect data in the literature to the donor that they came from based on matching up the identity based on genotype. So that's been extremely useful to try to connect data. And again, we're a bit surprised by how oftentimes there's no RIDs, or there's no like direct link from the donor in a paper to the source that it came from. It's also useful for identifying sample swaps, which in any context, in any study, no matter who you are or what you're doing, probably happens like 5 to 10% of the time. So it's also very useful for identifying that. Okay, and then a lot of documenting the processes and standards, so metadata, how it's collected, data processing, quality control, as well as we thought quite important summaries of data in terms of key metrics, maybe help people identify outliers or patterns that we wouldn't have noticed, and just give people access to summaries of this information so they can decide how they may want to analyze it. Okay, I'm almost done. So yeah, and I think taking these resources that we're creating and in many cases collecting and organizing, and then providing this to users in a variety of ways through both browsing data, querying it, just downloading it, or even just access to buckets that have this information organized directly, I think is actually quite valuable. But then also more advanced things like analyses, libraries, workflows, and other tools that sit on top of these data, as well as providing all these standards. And also, I think important to highlight the sources of data that are providing this information to PanKbase. What are the strengths of these resources? What type of information they provide? What benefits do they have to researchers? Okay. So I think there's maybe two major keys to the success of the platform long-term outside of developing the platform itself. Both concern outreach. And so one is engagement with the data providers, the repositories that contain these data, as well as experts in the field, and to leverage their knowledge not only of the data that they're generating and collecting and providing, the metadata that they're collecting, but also the insight and knowledge of expert investigators. So I'll just give one example, but there's many of these. So if you're looking at, say, a single cell map, annotating the identity of different cell types or subtypes, they even have like immune cells, there's a lot of value in tapping expert knowledge of what immune subsets exist in the data that you're looking at, right? So examples like that. And then also outreach with the community. So better understand the needs of the community, tailoring the development of the platform to help serve what people actually need, not assuming what people want to do, but like actually understanding what it is that the majority of users are wanting out of a platform like this, and then trying to develop the platform accordingly, as well as opportunities for people to contribute back to the platform. So on that note, so we have a survey that we're releasing to help understand needs of the community. So if anybody here is interested in filling out the survey, you'd be the first ones to fill it out. So I'll leave it up for a minute. I think based on the results of these surveys, then that can help us understand where our areas of focus should be, and also like what types of tools and things that people might be interested in. So I'll stop there and summarize that again, that this project just started this year, but we're excited about the progress we've made so far, and then also excited to have a very early release of the platform this coming January at the HERN 10-year anniversary meeting. So thank you very much.
Thank you. The three speakers in this session. So that three speakers are representing NIH-funded PIs that generating data, provide the platform, curate the data for the community doing AI research. Next, I will introduce my colleague, Dr. Rebecca Rigridge, who's the director of the NIDDK Central Repository. That is the resources that actually host in our NIDDK. Rebecca.
Everyone. Can you hear me? Now, better? Good morning, everyone. I'll try to be louder and project my voice a little better. Bear with me, I do have a condition, dysphonia. Okay, so to get started, thank you again. It's very nice to be part of this conversation. So to start, as we have heard throughout this workshop, AI-driven data science is transforming opportunities in AI-based in precision medicine by enabling personalized treatments and promises to continue to provide opportunities and improving treatments and promises many more. So to improve healthcare outcomes. I want to focus to this conversation on 3 critical aspects from a repository's perspective, and those are data, infrastructure, and I'm sorry, I'm losing my line here. Data infrastructure and innovative and easy to use tools. There. So following the slides, I'm gonna briefly touch on the resources we currently offer and that we're developing to achieve AI-based data science and potentially improve secondary research. So for those of you not familiar with the Central Repository, we are one of 27 NIH controlled access repositories, and we were established to expand the usefulness of NIDDK funded research. We have three overarching principles. Those are to improve the quality, increase the impact, and advance the understanding. We accept resources of NIDDK funded consortia. Mostly, those are multimodal data and their associated biospecimens. Dr. Germino made reference to that earlier. Our commitment is to expand the usefulness of those resources. And we have, last year, we committed to renew our commitment and expand those into specific areas that those were, like you see here on the right, to commit into innovation, process improvement, system usability, assessment and accountability, and NIH policies and initiatives. Represented in these graphics are some requirements that we have identified and some enhancements we began implementing for AI-based data science within these areas, from adherence to NIH policies and initiatives that I can mention, all that map back to the core principles I mentioned of data, infrastructure, and easy-to-use tools. At the core, we have data. We all know that we need good data. And to make data reliable, we need to focus on data that is accurate, reliable, high quality, and interoperable. We also need to understand that this is not easy, especially when it comes to legacy resources. So the Central Repository is proactively working to increase the impact of the resources we receive, and also to make sure that we continue and make educational effort to reach that approach. So recently we implemented a metadata approach to standardize, validate, harmonize, protect, and correctly annotate the resources that we have and receive. We are also establishing a data challenge series that is aimed at developing solutions for making NIDDK datasets AI-ready, for which I will speak a little bit more in the coming slide. Next, to unlock the potential of AI, we need to create a more scalable, secure infrastructure that is flexible and interoperable with other systems. We also want to ensure that the safety and the reliability of the data we hold. To achieve this, the repository is continuing to modernizing our infrastructure, and we are leveraging opportunities to streamline our operations and to improve compliance with NIH guidelines. For example, we're gradually right now transitioning our system functionalities to a metadata-driven system architecture model that will pave the way into advanced analytics and discovery tools. Those are hopefully will be fair, scalable, flexible, and be secure and easy to use. Speaking of easy to use, we are finally need to have those that are validated and that enable translation of data into actionable insights that promote also the collaboration, sharing, and partnerships from again, from a repository's perspective, increase the pipeline of secondary researchers. One of the things that we're looking to do with our platform is streamlining end-to-end data science lifecycle and discovering that will lead to the discovery of data-driven biomedical insights. For example, last year, we started implementing a piloted analytics workspace and are now implementing the lessons learned from that time to deploy a cloud-based analytics environment where clinical researchers and data scientists can access a suite of resources and analytic tools that will help us bring that to change. We are also looking to conduct other data challenges and AI innovation. Hopefully, the analytics workbench will streamline, like I mentioned, the data science lifecycle and facilitate these biomedical insights. Other anticipated benefits of this is the promotion of collaboration, supporting innovation, and minimize the data movement. I am going to take some time to make a plug for the data science-centric challenge series. And those are, I can mention, one of the approaches or hopes that we have for this data challenge series is that we want to develop approaches, models, and methods to increase data interoperability for the resources that we have. We want to use those resources for machine learning applications. We also want to make sure that we enable the discovery of innovative approaches. So feel free to come onto our website and visit for additional information on the data-centric movement and to join. We are also going to start in January 2025 a secondary data science and meet the expert webinar series that will have a number of topics. They're going to start from data science fundamentals to artificial intelligence, fair data sharing, privacy protections for sharing human research data, as well as different participant privacy techniques, other challenges and opportunities and considerations for secondary use of electronic health records and real-world data resources. I'm also including here the link for past recorded tutorials and webinars and other educational resources that are available on our website. So please check the web page frequently because we're going to continue to update as that becomes available. And with that, I want to thank all the support teams. So if you have additional questions on the analytics workspace that is being developed, all of the resources that we have, please, our support team is here. For the technical questions, go to them. For the programmatic questions, you definitely can ask me, but also feel free to ask any questions. Thank you.
Next, let's have all speakers come to front.
How long do we have for questions?
Yeah.
Shorten it or?
We need to.
Yeah.
Right.
Right. You want to come to the microphone and ask your questions? That'd be great.
Hi, I'm Chuyan from.
Let's turn the mic on, yeah. Thanks, John.
So, I have a quick question, but probably, to answer this question is, a lot, the complex. So basically, my question is how to incorporate the genetic information into the space in your comfort zone. So to address, this question, I think, I suggest two very important aspects. We are recently observing. One is the enormous new individual whole genome sequencing data. For example, UK Biobank and all of us, and Vanderbilt also has our ADD project combining almost million individual whole genome sequencing data. So it challenges you not only just in volume, veracity, it's a big problem, but so many things in knowledge. So, this is one aspect. The other aspect is genetics is now also seeing this change with very different kind of, paradigm shift from monogenic to polygenic, even in omnigenic. So it's not just simply, looking at genotypes, or something like, the single tissue, or something like more in a broken down system you are familiar with. For example, Knowledgebase, for example, simple phenotyping, but variability across individuals so huge, right? So these two aspects a challenge in the entire community. So I wonder how you actually consider this opportunity, and also challenges from your actual background and what you are working on.
I guess I can answer that to start. So yeah, I think in terms of how you might consider using genetic data in light of all this other information, a couple ways that we have been thinking about it, both like in the project, but then also in our own research, are one, trying to find ways to summarize an individual's risk, say like polygenic risk scores, even overall, or perhaps subset it into maybe heterogeneous groups and polygenic risk scores within groups, and then asking whether there's a relationship between polygenic risk and other molecular phenotypes or clinical phenotypes or whatever was collected from the same individuals. So that addresses in part like the polygenic nature of both type 1 and type 2 diabetes, outside of just looking at individual variants, but looking at like somebody's overall summarized risk. But then I also think too, just even looking at individual variants, their relationships to molecular readouts and how that potentially informs diabetes risk and identifying individual genes to study, for example, I think is still very interesting to many researchers. How you handle the large volumes of data, I mean, I guess in the pancreas, it's less information. There aren't as many pancreatic donors that are available, and many of those have genotyping data available. We're not talking about like millions of individuals. It's on the order of thousands. So, if you're asking questions that relate genotypes to data collected in the pancreas, I think it's probably a more tractable problem. But yeah, I think how that then relates to information that's collected, say, in biobanks, some of which you mentioned, they have great platforms to analyze data. I mean, all of us, we use all the time, and I think is a phenomenal platform for analyzing data that's present in all of us. And you can upload your own data and use that data along with information in all of us, which we have been doing increasingly as well. So I think that's probably one way to connect the types of things that we're doing in like the pancreas space to larger biobanks. But yeah, others have.
I can try to kind of give like a more naive aspect. I think of it as genetics as one of the many factors that are contributing to the phenotyping. So not just the only one. So we know that a lot of environment, somebody mentioned yesterday about diet and, exercise and all that. So how do we integrate all the complex multimodal data and then essentially really pull the data, create a data that's big enough and use sophisticated machine learning to really generate a hypothesis. So that would be an interplay between genetics and other risk factors that you are able to integrate. And I'm hoping that after that, you're gonna, have to create a validation study as well.
Yeah, the whole genome sequencing provides you entire genome-wide predisposition. It's not just part of your information. It can connect everything to what you are working on. For example, how different people with your phenotype data set with AI-READI can be explained in a population level. So it's challenging, just not only just different type of information, but it really challenge to AI, how we understand entire disease. So that's what, my, the questions in point. I don't think this is a new information, but it can be a good opportunity to integrate everything, but we don't not ready yet, even in conceptually how to consider that.
Al, do you want to go next? Yeah.
That was a terrific session.
Can't hear you.
Course, Vanderbilt University. That was a terrific session. Thank you for all four speakers. Dr. Lee, I'd like to ask you a question. I was intrigued by your computational core, that dkNET. It seems like if you build it, they will come, and they will be many, many, many people. So is this being viewed as a community resource that would address some of the challenges that people have in institutions about the computational? Tell us how you're thinking about that in terms of the scalability and the accessibility. Thank you.
The short answer is yes. A longer answer is there are a lot of challenges related to scalability and sustainability. And one model we are looking, a few components, AI is a key part of it. We want one reason this dkNET portal got renewed is to have the vision of bringing AI to the community. We want to make that process easier, smoother. The other aspect is really about this, really about scalability. I mean, if our goal is to make more people come to the platform and use the platform to do their to address the pain points. But the other, in addition to the software development and also the technical side, there's also side of the resources. On the resource side, my view on this is, if you look at the trend in the industry, a big trend is to move everything to the cloud. This is the trend. And there are quite a few very successful companies. I name a few like Snowflake and Databricks. What they do is they provide their infrastructure, but the additional computing resources are not provided by them. They go to this basically big three, really the Amazon AWS and the Google GCP and Microsoft Azure. So what they do is they have this platform on which you can do analysis. But if you do a lot of computing, need a lot of computing resources, you go to the big three to ask for resources. So our vision is the one I showed you here is more like the layer we call it brain. So that the basic features are provided on this layer, like user login, sharing of data sets, and workflow editing, and many other things. But for many computationally expensive tasks, we, including NIDDK, NIH, I don't think we have enough resources to provide all the CPU, GPU, memory, disk, the whole community. In that aspect, I believe the right model is to rely on the public cloud and let each individual user pay for the resources they use. So this is we are working on to run that direction. I hope I answered your question.
John?
John Cadiz, City of Hope. So Dr. Lee, just a quick follow-up for you, and I have one other comment question for Kyle. So you can bring workflow. So if I have something like Nextflow, I can bring it there?
Yeah, that's a very good question. This is one of the technical challenges we are facing. For example, in the workflow I showed in one of the slides, that workflow was originally provided by Professor Chen from Cornell, and also Sally in the audience. Okay. They provided the original program in R, and my team worked with their team very closely to read their code, understand the code, then convert the code to a workflow. That process took roughly about 1/4 or 3 months. But this is one of the technical challenges. And one of the technical issues we're working on is how to make the whole process very efficient, to minimize the effort of doing migration. But a lot of details we can talk offline, but these are all of the issues we are working on.
Okay, so a comment/question, Kyle, that we were talking about yesterday is connecting data from the literature is going to be very difficult, dare I say impossible, even using AI, if investigators have not identified the individuals that they're getting the data from in a protected way. So you like you referred to RRIDs, for example, and like using the IIDP example, dating back to 2009 all the way to date, every single one of those donors are identified. They have RRIDs, but when we go to publish these papers, and you've referenced, even given these standards that people have, people will not identify, generally speaking, some do, but don't identify the 20 donors using the RRID. So any amount of wonderful, terrific work isn't going to help connect that if it's missed up front. I mean, you're going to have to contact the author, and just at scale, it doesn't seem to work very well. So your thoughts on that, because what you're doing is you're essentially taking it from the aggregator point of view and addressing this problem, which is a really good way to do it, but still at the literature side, it's very difficult.
I would say, so at the moment, it's maybe somewhat tractable to like contact the authors of all.
Yeah,
because they know that.
They know
RIDs from owners, like when it's sent, they just know it's like, it's not a criticism. It's just, people don't always pass it on in the papers, right? So you could contact people, and if they respond, then you could collect that information. So that's a short-term answer.
Yeah.
And that supplements like genotype-based things, but that also requires that there's sequence data where you can extract genotypes to be able to match it up. I think probably the longer-term solution is just promoting and encouraging investigators to, and maybe even requiring from the journal end, this level of information. And all it really requires is those ID, like this.
Right. And a lot of journals do that. They do require it, and things are getting much better. So the point overall is that, I mean, because what you're dealing with is tractable, given that you found 300 data sets or so, right? So you can contact them.
It's tractable maybe in like the pancreas.
Right, exactly.
Extrapolated like any type of owner, and like any type of, I mean, anything of interest, even NIDDK, right? That may get a bit unwieldy, right? So I think it's, probably longer term is promoting people to, and probably requiring that level of information when papers are published, so that they can be connected back to the donors.
That's just the point I wanted to make, is that the tools are there. People in this audience have published on what these requirements are. And it would make everyone's life a lot easier.
Some journals, so like diabetes,
exactly.
Journals have requirements in that vein, but then, people publish papers in non-diabetes journals, even though they're using islets, right? And those are the cases where it's a bit trickier to align, but, I don't know how you would do it otherwise. Right? I mean, in some cases, maybe there's enough metadata provided, like age, sex, BMI, and maybe like where the hospital and stuff, but you could maybe connect it. But again, that's maybe you could automatically call that information. So that's maybe something could be a bit more automated. But I think the long-term answer is people just connecting it themselves.
Thanks.
Shubing?
Shubing Chen from Weill Cornell, and I want to thank all speakers, including two of my close colleagues. Very nice presentation to introduce the DK data science program. I have a question about the new technologies, because in last several years, like the spatial transcriptomics, like long-read whole genome sequencing, like long-read single cells are start to be applied to many models, including like pancreas, the organ we are very interested in. So I'm just wondering whether the speaker can give some introduction about the ongoing effort, or maybe some plans to incorporate all these new technologies, new data sets in the current NIDDK data science program.
Pardon. The central repository does have the ability to support many different data types. However, there's an eligibility threshold for submission of resources. If we mostly accept resources from consortia groups. So if consortia groups are developing these resources, they're most likely going to be accepted. We also accept other smaller grants that are associated with or that have significant scientific value for NIDDK. And those are evaluated post hoc mostly. But we do accept different data types, including single cell data, so long as it's flood files and things like that.
Yeah, in next several years, a lot of this data will come out. I think it's something worth to put in consideration. Yeah.
Yes, definitely.
Jake?
All right. Yeah, thank you. First of all, great job. I think all of your efforts are making this field from a data poor science to data rich science, and it's very promising. I promise I bring this to my community. But my question is more related to how to actually shape up our training to match up with the resource that we have come up with. Right. So the traditional way is actually someone mentioned that if you build it, they will come. But if you build it, they will come is based on the older model. They can still somehow manage in a Google search like interface and then say, oh, my favorite genes and get the data and leave. Now it's no longer the case, right? You have very fancy pipelines that maybe us data scientists can understand and use. And then your data have nuances, what it can do. And more importantly, a lot of the diabetes researcher will say that they have models that you don't have. They have knockout genes and then conditions that it doesn't cover. But it's not easy for them to actually figure out what resource to use and what they can do to add value. If they have $0, what can they do? If they have 10,000, what can they do for experiments? If they have 10 million, can they actually make a drug out of it? I wonder if you can actually comment whether this is your job or this is something that we need to work on and think about or outsource to ChatGPT. Thank you.
I can start that conversation. I don't agree with you build it, they will come. Let's start with that. We do have the central repository. The central repository is a free resource for the most part. And they don't come. The resources are there and the utilization is not up to par. So you build it, they do not come. So there's a lot of outreach that we need to do, constant outreach and education to let people know how things are to be used. So to your point, things need to be easy to use and you need to explain how to use and how usable these resources will be for their specific science. So maybe others can comment as well. It's not just simply build it. There's have to be that outreach, constant, progressive build up and enhancing the resources so that other people can see how easy they are and moving and constantly being flexible and adaptable to the new technologies as well.
I would agree.
I can comment on one of the large meetings for a Bridge TI program. What we did was we had like an open house for various data generation projects, and then it was a mix of kind of high level discussions of data design and study designs, but then also we had more of a data-thon type where all the machine learning AI specialists came in and they were just, exploring the data. They were able to download and then create those models instantly. So we created some of the challenge that they could work on. And so I think workshop like these where we present the data that exists and then we could troubleshoot and then think about, is there any connection that we could tie together between data sets, but then also a real-time workshop where data scientists can actually, get their feet wet in the data set, because I think it's just, hard to imagine unless you actually have the data set and then there are questions that are very applicable.
Regarding the question about after you build it, do you come? I want to make my own comment. I think a fundamental question here is whether what you are building is useful. I think if you're really addressing a pain point of some users, then they will come. But if what you are building is not useful, I'll mention the usefulness shortly, then no matter how hard you push it, people come, people leave. people, it's easy nowadays, it's very easy for people to find it, to know it, either through search or is this kind of social events. That's very easy. But when they come and they really enjoy what you're building. So now there are two aspects of this usefulness. First, whether you're really solving their fundamental problem, whether they have a pain, not just like I'm not delivering a vitamin, I'm delivering a painkiller. So it has to be a painkiller, people will come. Second, when they come, your system has to be easy to use. Both are very critical. Now, when they're easy to use, many things like the interface has to be very straightforward. I mean, this idea of good example is Facebook versus MySpace, if you remember the old days, right? So both are social networks, but one succeeded because it's easier to use. Second one is you need to provide enough like documentation, tutorial. When they come here, they know how to get started very easily. That's why I believe usefulness, whether you can really deliver a pain point, that's the key to the question. Regarding the fact people have different amounts of money, I think depends on really how big each sector is. But if the person doesn't have any money, but they can still come to a place to get some value, that's how Google works, right? People can still get something. But for some people with 100K, they might be able to get something done. And if there are lots of people in this category, then you can still have a kind of a mini market. But for the people with $10 million, they may just have their own solutions. They might not go to you. So it really depends on the size of each group of people.
I love this discussion. We maybe can save it for the break. But one more question, Ronald, do you want to go?
Thank you. Ronald Ma, Chinese University of Hong Kong. Thank you all four speakers for your excellent talk and also the collective effort to generate these very important resources. My question is for Dr. Cecilia Li. So thank you for sharing this invaluable resource, the AI-READI dataset. I wanted to ask about transferability, or how much, to what extent is the data agnostic of the platform being used? So of course, exposome data is exposome data and retinal photo is a retinal photo. But for the OCT measurement, are these standardized metrics that would not be affected if sort of different equipment is being used to take the OCT measurement?
So as you know, the device, so the lack of harmonization between ophthalmic imaging devices is a huge problem. So really to tackle that problem, we are scanning, like for instance, OCT, we're scanning through 4 different devices. And so they actually go through 44 different protocols of multiple devices and then different modalities. And it is our hope that there can be created like AI models that can do transfer learning, and then also kind of impute, can you do like a generated AI model where now you have the data from, let's say a TopCon device, can you generate models that looks like images from, Heidelberg, so things like that. So we have worked that in the system, and then so hopefully, smart researchers like you, can leverage that and then create those models, because it is a huge problem. And we've also created a lot of DICOM standard, and then did a lot of, standards work so that new imaging modalities such as FLIO, we were able to create DICOMs for them.
Thanks very much.
This is great discussion. I still see a lot of questions coming. Let's address this in a break, maybe. And online questions, hopefully each speaker can help answer those questions online. So we can get back to the time, because I know lots of people have flight to take, so we really cannot push back too much. Thank you, everyone. Thank you for the great discussion.
All right, so it's my distinct pleasure to welcome Dr. Eran Segal from the Weizmann Institute in Israel. He's a professor in the Department of Computer Science and Applied Mathematics. And his lab works on a number of things, and it's a group of computational scientists as well as more bench type researchers. His lab works on 3 main areas: personalized medicine, use of big data in healthcare, and the role of the microbiome in health and disease. Unfortunately, we're not going to be able to hear about all those topics, but today he's going to focus on the human phenotype projects, personalized medicine based on deep human phenotyping. So thank you.
All right. Hi, everybody, and thanks for the invitation to speak here today and tell you about our work. So I want to tell you about the large-scale human cohort that my lab has been developing in the past 6 or so years, and the large-scale data that we're collecting, and then the AI tools that we're developing on top of that. And I'll focus more also on the continuous glucose monitoring work, obviously more related to this meeting. So what motivated us, I don't think we need introduction to motivation for large-scale cohorts, but really, if you look at what happened since the publication of the human genome in the year 2000, we went from knowing just a handful of genes involved in some diseases to where we are now, where we know hundreds and thousands of variants involved in virtually all diseases, given the GWAS studies, and use of genetic data in development of drugs basically increases the chances by twofold. But of course, the genome is just the genome. It doesn't take into account environmental factors, everything that happens to us during life, exercise, nutrition, and so on. And for that reason, we established the Human Phenotype Project, where the idea is to measure, in addition to the genome, all the other phenotypes that we can. And so this is a slide that shows all of the types of data that we're collecting. So before people come to the clinic, they go online, they fill out hundreds of questionnaires, basically everything that's on UK Biobank and some additional questionnaires, medical background, family history of disease. They upload all their electronic health records, historical blood tests. They then come to the clinic. They do various body measures, blood tests on site to standardize between people, hand grip strength, ECG. We record voice. We have various imaging modalities, DEXA for full body scan, bone density. We use high-resolution images of the retina to look at blood vessels for cardiovascular disease, ultrasound to look at fat in the liver and the carotids, also for cardiovascular health. Then we have all the multi-omics that we're doing, so whole genetic sequencing. We follow vaginal and gut microbiome, metabolomics targeted and untargeted by mass spec, proteomics from blood, RNA sequencing from blood. We have a novel immunoassay that we developed, actually adapted from Steve Elledge's lab, that basically can tell you your entire infectious history. Then people go home with sensors for tracking physical activity, for monitoring sleep during 3 nights with various sensors. I won't have time to discuss. Continuous glucose monitors for a sensor of 2 weeks. During that time, people log their diet, medication, physical activity, and we biobank PBMCs and serum. This we've done so far on 13,000 people in Israel. It's a longitudinal cohort, so every 1 year we contact people virtually and get an update on their medical status. Every 2 years, they come physically and undergo all of these tests, except for genetics, of course. Some of this data is really kind of novel and unstructured, like what I'm showing here. We have some gait data that people do these various tests of walking. We video them, and we can then extract basically their skeletal positions, and this is another type of data that we have here. Overall, this data covers around 17 different systems of the body. There's about 40 different diseases that people have. It's the general population, so pretty typical of what we have in Israel, and also similar to what is here in the US. About 30 medical conditions, and seeing the usefulness of this, we're now in the process of trying to expand this. The next milestone we're going to try and hit is going to be at the scale to 100,000 people, which we hope to do both by increasing the cohort in Israel, but also by opening clinics in other countries. And in Q1 of 25, we're going to start a clinic in Abu Dhabi and another one in Japan, and hopefully we'll be able to roll this out to other countries. The UK Biobank was mentioned here. I think, in my view, perhaps the most impressive cohort that really transformed biomedical research. Very impressive if you look at the number of publications, over 10,000 from 40,000 researchers in 100 countries, major investments by pharma companies to basically enhance the data. So in comparison, we're obviously not as large as the UK Biobank, but I think we have deeper, broader phenotyping, and it has several unique characteristics, the ones that I'm showing here. First, we're going to be the first cohort, as far as I know, to be international in multiple ethnicities, genetic backgrounds, cultural diversity, and so on. And then all the other measures here, maybe most interestingly, the state-of-the-art multi-omics like the microbiome RNA sequencing, untargeted metabolomics, and so on that we have here that is not part of the UK Biobank. And the data can be accessed. You can go onto the humanphenotypeproject.org and request access free for non-commercial academic use. Over the years, we've been publishing several works on this. But hopefully now that we're giving access to other researchers, we're going to see a larger number of publications come out, much like what happened hopefully with the UK Biobank to increase the impact of this cohort. So let me tell you about kind of from a high level some of the projects and discoveries that we've made on this cohort. And actually a prelude to this cohort was a smaller cohort that we established. We called it the Personalized Nutrition Project. This morphed eventually into the Human Phenotype Project. This was this work we already published, so I'll be very brief. But we were looking at personalized nutrition, combining gut microbiome data. And what we showed is that when different people eat exactly the same meal, they have very different blood glucose response to food, as shown here anecdotally for our participants, but we saw this on the scale of 1,000 people. And you can see different people represented by different colors have different responses, but the same person eating the same meal, in this case, 4 slices of bread, has very reproducible responses across different mornings in this case. We were then able to take clinical information about people, microbiome data, and develop an algorithm that quite accurately predicts these glucose response to foods. Here, every dot is a meal, and you can see between the y-axis and the x-axis, the predicted versus actual measured area under the glucose curve, showing a pretty highly accurate results. And then based on this algorithm, we could design diets tailored according to the algorithm that take people who are pre-diabetic. You can see these spikes in glucose levels during the course of a week, and then by tailoring a diet to them with the algorithm with exactly the same amount of calories in every single meal, we could fully balance the blood sugar levels of people. This was part of the first paper we published back in 2015 on a small cohort. This intervention was done on a small cohort, and then we went on to do a randomized clinical trial. It took us several years, where we took 200 people with prediabetes, randomized them into either a standard of care Mediterranean diet or the algorithm-based diet. The intervention lasted for 6 months. Here's an example anecdotally of one participant on the algorithm diet arm. This is at the top 1 month of glucose monitoring. You can see many spikes in glucose levels. And at the bottom, you can see the same participant during the first month of intervention, basically fully balancing the blood sugar levels of this pre-diabetic participant. And looking kind of statistically at all of the results, at the center, there's the hemoglobin A1C changes, where on the Mediterranean diet, we saw some significant reduction, but double the reduction on the algorithm diet. And it was even much more dramatic when looking at the CGM. We actually put CGMs for 6 straight months on all these participants, and we saw a major reduction in spikes in the CGMs after meals on the algorithm-based diet compared to much less reduction in these spikes on the Mediterranean diet, and also improvement in many other metabolic parameters. This we published a couple of years ago. So this for us completed a journey of about 10 years, going from an idea to developing an algorithm to really doing a RCT that in our view is really a treatment for people with prediabetes. We also have another study, smaller scale, that we published on people with type 2 diabetes, also showing major beneficial outcomes in terms of glycemic control. So we have also in the group a major focus on microbiome. So I want to share with you also some of that work that we're doing. And typically in the field of microbiome, because mainly of the scale, the relatively small scale of cohorts, people have been mostly working on the composition of the bacteria, so which species are present, but afforded by the larger scale of participants that we have here, we are starting to go at a deeper resolution and really analyzing not just which bacteria are there, but looking at the actual genome of individual bacteria and basically doing like a GWAS in human genetics, but now we're looking at individual nucleotide positions across bacterial genomes and looking at how that variation relates to host traits. So the idea is that we take these metagenomic samples, we get these short sequencing reads, we map them to bacterial genomes, and then we record the variation at each position across people. And you can see at the top here highlighted in blue, there's some variation where the top participant, it's actually misaligned here, but because of, I think, font issues from going to Mac to PC. But anyway, the idea is that if we see variation, for example, the top participant on the next position has an A at this position and a high BMI, whereas the other two have a T at this position, but lower BMI, that would be could be an association between genetic variation at one position in a particular bacterial genome species and host trait, in this case, BMI. So this is obviously just an illustration on 3 participants. We do this on the scale of our entire data at the time that we did this, and this analysis that we published last year, we analyzed 7,000 participants, a large-scale cohort in terms of microbiome data. We identified over 1,300 different bacterial SNPs, so individual positions that had significant associations, in this case with BMI. What you're seeing here is a standard Manhattan plot, except that the x-axis is now not human chromosomes, but bacterial species, and still every dot here represents one position in one bacteria. The y-axis is the minus log p-value of the association with a BMI. So you can see we get many significant associations even after Bonferroni correction. What's more interesting, I think, is the effect size of these associations. So here's the same data, but now plotted as a volcano plot where the x-axis is the number of BMI points statistically explained by this variation at particular positions. The y-axis is still the minus log p-value. You can see some of these associations coming up as explaining 1, even 2 points of BMI. That's a huge effect, bigger than even the largest effects found from human genetics, even though those are done on the scale of now hundreds of thousands of participants. To me, this is not surprising because we previously showed a few years ago that there's a very little connection between human genetics and microbiome, meaning that microbiome is an environmental factor, and being an environmental factor, we know that environmental factors can have huge impact on traits like BMI. And here's just to dive deeper and show you one example. This is one of these significant SNPs, and if you just separate people according to participants that just didn't even have this species, so could not be analyzed, to participants who had one of the alleles at that position, the major allele, 2,000 of them, and then 400 that had the minor allele, you can see that just by separating them by the variant that they have at this one position in one bacteria, we separate 2 points of BMI between these participants. Here's another SNP explaining 1 point, 1.3 points of BMI. This is in an energy metabolism region, so we might hypothesize a mechanism. Here's another one, a SNP in an LPS modifying enzyme, so maybe related to inflammation, and we might also hypothesize a causal mechanism here. But I would say for many of these, we get, like in GWAS in humans, significant associations that we still don't have the mechanistic rationale to explain them, and that will be interesting to do in further research. What we are now doing as a next step is trying to isolate the relevant bacterial strains from individuals, so say from lean individuals, identify the strains that we see associate with a lower BMI, and grow them, and then eventually go into clinical trials where we would give them to overweight individuals and see if they have benefits in reduction of BMI. Another data that we have in this cohort is that we find to be very useful is the metabolomic data. So we do both targeted and untargeted metabolomics. And this allowed us to ask a very fundamental question, which is what determines levels of different metabolites? So because we have such a rich data in terms of genetics and diet and microbiome and so on, we could analyze each metabolite and see which of these different modalities that we collect on people best explains each metabolite. And we find that for some metabolites, it's the microbiome that explains them. You can see these associations, these high correlations on held out samples by a machine learning algorithm trained only from microbiome data, whereas for others, it's a diet that explains them, and for others, it's genetics. So with this, we kind of can build a database that for each metabolite identifies possible determinants of that metabolite. And that's important because we can then go and analyze disease cohorts like this one of acute coronary syndrome patients and take even just a single patient, let's say a 60-year-old male that underwent a heart attack. And we can then go into our healthy cohort and find matched controls for this patient, so 60-year-old males who are healthy. And then for each metabolite, we can build a distribution of the reference values we expect for that metabolite. This is a personalized matched reference control for this patient, and we can then see where the level of the metabolite for this patient falls. And you can see in this case, it falls at a low value outside of the distribution. And we can do that for all of the different metabolites and basically build a metabolite deviation profile specific to this patient, and then cross it with the database I just showed you that we devised to see what factors seem to be the ones that are determining the deviations in this participant. So are the deviations in metabolite for this individual, for this patient, driven by diet or microbiome or lifestyle factors and so on and so forth. And when we do that, we find that in some cases, two individuals that present at the clinic with the same condition, say they both underwent a heart attack, they both are 60-year-old males. When we analyze their metabolite deviations, they're actually different, and the underlying factors are different. So in one participant, it could be metabolites that are mainly driven by diet, and in another, metabolites that are mainly driven by microbiome, suggesting perhaps a different pathogenesis of disease and different treatment modalities that we might want to give to these patients. Kind of looking more holistically at the data, looking at many different modalities, we studied also aging in this cohort. So going back again to the metabolomics data, we could take only the metabolomics data and develop a machine learning algorithm that tries to predict the actual age, the chronological age of a person. And these are the predictions. You can see they're fairly significant, but not perfect. And not perfect means that individuals here at the top are individuals that when the model looks at their metabolomic data, their data seems like somebody more similar to somebody who's older. And conversely, individuals here at the bottom, when the model looks at their data, their metabolomic profile seems like more similar to somebody who's younger. So we can then ask if indeed individuals that seem to have a higher metabolomic age compared to individuals that have a lower metabolomic age, according to the model, whether that actually carries any meaning. And indeed, when we now overlay data that we didn't use to develop these biological clock models, as we call them, we can see that individuals here at the top that have a higher metabolomic age are also in worse clinical condition. They have higher BMI, higher triglycerides, hemoglobin A1C, blood pressure, cholesterol, and liver enzymes. So they're clearly in worse health condition, and we might identify by this markers of accelerated aging. We can do this for other types of data. So here's a biological clock that we developed from the RNA expression data. So again, individuals at the top, their expression level from PBMCs seems more similar to somebody who's older, and the opposite for people at the bottom. And again, here too, individuals with accelerated expression age are in worse clinical condition. Here's the retina scans. We can extract features and see how they vary with age. And overall, we see, as I showed you now, for two of these biological clocks, we see that higher age in a certain clock is actually higher risk, associated with higher risk for disease, and typically not just any disease, but a disease or the body system on which the clock was developed. And finally, in this work, we also see a difference between males and females. So with males, this is the metabolomic clock. Again, we see kind of a linear increase with age, but in females, we see this abrupt change right between the age of 50 and 55, which we could link to the menopausal state of women. So if we compare women of the same chronological age, but post versus pre-menopausal women, we see that post-menopausal women for multiple of these different clocks actually seem to have accelerated aging. So this was kind of a bunch of these different models and discoveries that we've been working on, and now I want to move to kind of the more recent work that we're doing on this data, kind of building more foundational multimodal AI models. And the general idea is that since we have so many different types of data, about 30 or even more different modalities that we're measuring about people, wouldn't it be nice if we could integrate all of them and build kind of a more holistic model of health and then use that to do various predictions and disease diagnoses. So what's fundamentally different about these types of approaches compared to the classical machine learning models that we and others have been building is that now we're actually using all of the data to train the models in what's called self-supervised learning. So the idea is that rather than just training machine learning models to do one particular task, let's say distinguishing people with diabetes versus healthy controls, we're now actually using all of the data, unlabeled data, to learn useful representations that could then be useful for other downstream tasks. So there's two main ways of doing this. One is called contrastive learning. So here the idea is that I can have, let's say, unlabeled data. So let's say I don't even know what these that these images are zeros, but I just know that they're similar to each other. So I would ask my model, could be a transformer model or any other type of model, I would ask the model to result in some embedding, some vector that represents each of these instances, where instances that are of a similar category, in this case the same digit, would be placed next to each other. But images that are different, like this 7 and 2, without even knowing the labels, I would want to push them apart. This is actually the basis of models like CLIP that align text and images such that you can then go and type text and get to see get an image generated that corresponds to the text. Another type of approach is next token prediction, which is actually the basis for large language models, LLMs, that are the basis of ChatGPT. So here we can take any sequence. It could be text, but it could be any other pattern, like even continuous glucose monitoring data, and you basically use all the history up to some point to predict what would be the next, say, glucose level. So these are the two types of approaches, and if you think about it, when you represent your data this way, you actually have a lot more data to train on. Because if we have, let's say, 10,000 people with a CGM that in 2 weeks has 1,000 different measurements, then we have 10 million data points to train on, as opposed to just one label for each of 10,000 people. And so with that, we actually developed a model that we call GluFormer on CGM data. And so we took, at the time of developing this model, almost 11,000 participants, and we built a transformer model, basically very similar architecture to large language models, where given the history of CGM data up to a point, you try to predict the next data point, and then hopefully show that it's useful for various downstream tasks. So first of all, just by after you run this model on the CGM of a person, what you get out of it is you get some representation. It's a vector. It could be of different number of entries depending on how large you make your embedding. So we chose 1024. So you get this one vector that kind of represents the CGM of that person, and then you can do like a standard PCA. In this case, it's a UMAP reduction to lower dimensions. And in this case, we can see that one of these dimensions actually captures fasting glucose. The other captures the postprandial glucose response. So this already shows that the embeddings that the model is outputting are actually capturing some useful axes, useful information about people. We can also use this as a generative AI model that, much like in text, you can type some text and then generate, complete a paragraph or even more. We can input CGM of a person up to some point and then ask the model to complete the CGM of a person, and it actually generates traces of glucose that seem plausible compared to the real data of the person. And then we can do various useful downstream tasks, like take the embedding and use it to predict various clinical measures, like the hemoglobin A1C or triglycerides levels or blood pressure or liver enzymes and so on. And these predictions of the model embeddings are shown here in blue compared to state-of-the-art, which is some package, it's called Igloo, that takes CGM data and extracts many useful, several dozens of useful features. And if you use those features to try and predict these measures, you also get significant results, but worse than what we can get with the foundation model on the CGM data. And it can also, we also show that it predicts not just clinical measures at the same time of taking the CGM, but also clinical measures, changes in these clinical measures 2 years to the future, and also 4 years to the future, better than the standard of the state-of-the-art Igloo model. We also showed the generalizability of this model. So we trained it on the cohort, the Human Phenotype Project cohort that we established in Israel, but then we extracted 15 external cohorts from 5 different countries, also using different types of CGM devices, also using different types of patients, like type 1 diabetes patients, which we did not have in our original cohort. And we see that in these cohorts, the model also achieves as is, but just by running the model and taking the embeddings, it also can predict clinical measures in these other cohorts. So that shows the generalizability of this to other devices, other geographies. Another thing that we looked at was looking at randomized clinical trials and seeing whether these embeddings can also be useful in predicting outcomes of clinical trials. So we went back to the RCT that we did on prediabetes that I showed you before. That's the one here on the left. And here, just by analyzing the CGM before beginning the intervention, the model could actually achieve fairly good predictions in what would be the outcome of these after the 6 months of intervention, better than what could be extracted from the Igloo features. And then finally, we also combined the glucose profiling together with diet to try and align, like you align image and text, to try and align diet with CGM data. And by doing this alignment, after we learn from the CGM of a person, we can then show that we can take only the diet data of a person and then generate what would be the trace of CGM for that person. And that corresponded quite well to the actual CGM of that person. So this is a nice application that after you learn from a CGM of a person and you learn from all the participants, then all the subjects, you can then only take the diet of a person, a diet logging, which is basically free, and generate a fairly accurate CGM profile. Another type of model that we developed just to illustrate the other type of learning, the contrastive learning type of approach, was one that we did on ECG signals. So here, what we did was to take the ECG signal that we record for 10 seconds from a person and divide it into the first and last 5 seconds of recording. And then what we asked the model to do is to align basically ECG of the same person, but the first and last 5 seconds, to align them together and push apart ECG signals that are coming from different people. And we can also do this across different visits. So if we have the longitudinal data, we want to align the ECG of a person from the baseline measurements and the measurements done after 2 years, and push apart measurements of different people. And this also showed first internal validation in being able to take just one trace of ECG of a person, and out of thousands of other ECG signals, identify which is the other ECG that corresponds to that person. And this model was also useful in downstream tasks in predicting various other types of data like cholesterol levels, creatinine, even RNA expression of some genes better than models that extract various features from ECG data. Another type of model that combines two modalities was for the cardiovascular system, where we aligned retina scans, where you can see the blood vessels and they're informative of heart disease, and carotid ultrasounds, also informative of heart disease. And here also we also used contrastive learning, where now we can do various different alignments. We can align the same from the same person different types of images. So the retina scan and the carotid ultrasound, we want to align them together because they're coming from the same person, and push apart images coming from different people. We also have images of the left eye and the right eye. So for the same person, we want to align those together. And then we can align across longitudinal visits. And this also showed good performance in internal validation and prediction of downstream tasks. We're just at the beginning of trying to build these models for the gate data that I showed you before. So here, kind of the version of next token prediction is basically to try and predict where the skeleton of a person would be, given that I've seen his movement up to some point. I think another exciting area of integration is to integrate actual large language models that presumably have read all of the scientific literature, which we never take into account when analyzing just the data statistically. So here the idea would be to basically take data and inject it into kind of use networks to convert it into the language that LLMs speak, and then inject it together with the LLMs who have information about the literature, and by that reach predictions. And in closing, I'll say that what we are working on now is kind of the more holistic model that hopefully will be able to combine all the different types of data. And the framework that we're using for that is also the framework of next token prediction, where the idea is that we take all of the modalities of a person and we put all of them, we consider each of them to be kind of a medical event that we put across some timeline. And so every time that a person comes to a visit, we have many different measurements that we take. Every time somebody reports some information about their health status, that's another medical event. It has a timestamp. And so we can look at all of that trace, and then basically use all the historical data of a person to try and predict what would be the outcome of the next medical event. The next medical event could be just the next glucose levels in 15 or 30 minutes, but it could also be which medication that person will take in a year, or what diagnosis that person will have in 2 years. When we actually put the data this way and we tokenize it by kind of across as a sequence of events, we actually end up with almost 20 million tokens per participant. So that's about 20 billion tokens, data points that we can try and do this next token prediction on in the entire data set. And so we're hopeful that this will allow us to learn useful models. And why are these models useful? Because once you actually have them, you can inject synthetic tokens, which would be correspond to interventions. And you can ask the model kind of as like a digital twin, you can ask it, what would be the outcome if that subject now lost weight, changed their diet, or had some pharmaceutical intervention, and get with that hopefully useful predictions. So as I mentioned, we're now trying to scale up this cohort. You can access it in humanphenotypeproject.org. And hopefully this is the type of data that will allow us to build the next generation of foundation AI models for precision medicine. And I'll just put this slide of my group who led and drove all these projects. And thanks for your attention.
So thank you very much. So this is now open for questions. While people are coming to the microphone, I'm going to take the prerogative of asking the first question. With your glucoformer, have you taken the steps to try to integrate not just diet, but exercise within the predicting, and how's that going if you are?
Yeah, so we haven't done that yet, but this is part of what we're now building with the model that integrates additional modalities.
Okay. Mike?
that they're food tracking and they're bringing round two. You have a food database, food tracking and bringing all this in, and how good is your what is it, 4 days of tracking, or it's the whole CGM time, or what exactly is that?
Yeah, so people actually, they report for about 2 weeks. That's the 2 weeks that they're connected to the CGM. They're highly incentivized because they get a report then of their CGM aligned with the diet, so they can see which meals spiked and which did not spike their glucose level. So they really want to know, and that's why they log, I think, quite accurately. And then some people just find the app to be useful, and then they just log even outside of the CGM. I think we have over 1 billion calories logged, about 500,000 days of diet logging. That's the scale of the data.
Great.
Over here.
For those foundation models you mentioned, did you train them from scratch or using some existing models? What are the resources and time needed for training each model, and are they publicly available?
So some of them we actually do not train from scratch. The ones that are image-based, we take a Dino or some other versions as pre-trained versions on images, and that actually helps performance quite a lot. For some of the others, we train from scratch because for CGM, for example, there's no available foundation model. I think our data is the largest for that. So those we train from scratch. Typically, actually, even though we have, I think, enough data and a lot of data, but tens of millions of data points, it's still, in terms of scale, it's enough to be able to train these on one GPU for several days. So that's typically how we train them. And yes, we are making these models also available. And so in addition to getting access to the data on the platform, there is also access to the models as we're developing.
Thank you for the wonderful talk. It's mind-blowing. I wonder, after you build this foundation model, are you also thinking about a collection of downstream tasks? And I assume the community will really welcome foundation model like this, and they will build many different tasks for their own problem. So are you also thinking about how to make a community or ecosystem of foundation model downstream tasks?
Yeah, so like in the answer to the previous question, yeah, the idea is to make these available exactly for that, because once we've shown that, as I showed briefly in the results, that these models actually generalize to other populations and to other CGM devices even, then what it means is that if you take these models and you just run them on a CGM, you'll get an embedding that contains a useful representation of the glycemic control of that individual. And then even if you have a small cohort, even 10 people, 50 people with CGM, those embeddings could be useful. And I can imagine many downstream tasks and even some that I can't even imagine now that people will be creative and develop. And we want to see that happen. So yes, we'll make the models available.
And you have used a large collection of data to train these foundation models. Do you also have plans to increase the cohort, or maybe using this ecosystem to further enlarge this cohort to train these foundation models?
Yeah, so yes, so the idea is that we'll keep training these models as we collect more data, and we hope to collect more data because we're trying to recruit more people, increase the size of the cohort, both in Israel, but also in other sites, and that all will be part of the Human Phenotype Project.
A follow-up question is, will you also be able to address different population, this underrepresented cohort, something
Yes,
like that?
for example, the next two clinics that we're opening are in Japan and Abu Dhabi. And for example, in Abu Dhabi, this will definitely be the largest cohort based on an Arab population, which is definitely underrepresented in a study. So I think that'll be very interesting and together kind of add to the diversity of the data.
Thank you so much.
Oh, go ahead. This is from.
Yes, I have a couple of questions from the online chat. Dr. Christopher Lynch is asking that gait has been used to predict Parkinson's disease before any overt symptoms. He's asking if you have applied those approaches to your gait data.
Not yet, but yeah, that's part of what will be, I think, very interesting to do.
Go ahead.
I'm happy to turn. How wonderful. Thank you so much. Your talk was fabulous. Deb Gibson, NIDDK. My question to ask you is whether you intend in your data expansion to include things like kidney biopsy, or really to analyze the data, not from a report, but from essentially the images, digital images from the tissue itself. If that's part of your plan, and if not, I'm just going to try to plant that seed to sort of expand that data source.
Yeah. To be honest, right now, that's actually not part of the plan, because the idea is to have a cohort that we track, which is kind of minimally invasive. So mostly blood tests, CGMs, but not, so we're not doing biopsies. But
not to do the biopsies, but to capture the data. So should that
oh,
event occur,
I see. So,
right?
so if
And
people, okay, yeah, sorry. So yes, so we have all the EHR of people, and yes, so ideally we would collect all the imaging data, everything that people have, and then you can try to align that with the data that we have.
We'll talk later.
Okay.
Okay, last question.
Perfect. Another question from Dr. Adam Garita. With the glue-former model, are you able to look at what the self-attention units have learned and if meaningful knowledge can be extracted from that?
Yeah, that we're in the process of doing that. This model we just put out about a month ago, but yeah, it's a very good question to then introspect the model and see actually which features of the glucose profiling it's mostly using to generate the predictions.
Thank you.
Okay, last question, short answer.
Okay, yes, excellent talk. So my question is focused on the aging and diabetes. We know that diabetes happens a lot in the elder populations. So did you ever try to compare that healthy aging with normal BMI with the aging and the diabetes to see whether they have the different metabolics being affected?
We're doing some of that analysis, but we don't have the results on that yet. But again, there's just all these questions are really good. There's so much that you can analyze with this data. We can't do it all, which is why we're actually excited to open access to this data to have everybody do and ask questions like the one you asked.
All right, well, thanks. Let's thank Dr. Segal for his presentation. So let's see here. So we're now Zhijing, we're now headed to our breakout group. So this will be like yesterday. We're going to break out into the 4 groups, and we'll be focusing on topic area 3 and 4 before coming back and kind of summarizing and hearing from what each of the individual groups. So there'll be 3 groups in here, and the purple group is where they were yesterday. Anything else? Oh, go ahead.
So for the breakout, we're going to 2 breakout room in this one, which will be the same position. The middle one, the orange, we will put on the poster room. So the purple one still in the next room, that is the classroom in there. So it will be kind of 4 groups. In this way, kind of less interaction for each other. So we will set aside so we can try to find your groups.
Okay, so again, orange is the group that's moving out of here to the poster room. Purple over there, and the other 2 groups that were here yesterday, one there and one there. Yeah. Thank you.
You're on the stage. Then you can choose.
So what is the time for?
So.
For online people, the session will come back on 12:45.
She's using Olivia. code and it was crashing. I read the code using the whole NAT model. And I was like, why are you using the NAT model? Why are you not using the NET model? But
try to finish in about 3 minutes each, and please focus on what you have to add or what differences you guys discussed. Just focus on those additions and the differences. And the four groups will take turns to lead the discussion. Yeah, thank you. Maybe we can get started with the first topic. Green, I guess the green team go. Topic 1.
Okay, so I'll start. And we first talk about the data challenge. Actually, we mostly talked about the data challenge. And so we were talking about the data centralization, harmonization, which is really good to see this morning that there is effort trying to do that. And we also noticed that there's some lacking of some modalities, especially diet, exercise, lifestyle. So hopefully this type of data will be generated and centralized and harmonized with everything else. So one idea we were talking about is that for healthy control, can AI-H request all the people who work on projects, any AI-H projects, to donate their data as a subject? And then we form a pretty good healthy control. May not be a little bit biased, but it can be a good healthy control. You want to do it next?
yeah, if you want Jeremy.
Push the button.
So yeah, so I think to add to that, I think during the discussion it came up that a really available AI ML ready data set, it does not exist yet. I mean, because there are so much concerns of using this that database versus that database. So a well annotated, harmonized, standardized database need to be established. The other topic that we went through is the difference in definition, right? For example, foundation models or basic terminologies. These are still missing, and how to combine foundation models among different data sets, or how to tune a foundation model for a given data set, right? Are we there yet, right? So it's a recent buzz, the foundation model. So question is, if I have to study a very specific rare disease, is that foundation model granular enough to capture that variability? The other thing we discussed is standardization of the data. So standardization, as we know, that it's a very difficult topic. And it was brought up that there are industry standards, at least for EHR data, which could be used for standardizing AI-ready data. But I work in image field, so there I'm not aware of much industry standard. So question is, what could be the standard format? And in HubMap, Human Biomolecular Access Project Consortium, we recently came up with the big list of standard data format. But question is, how much the community know about it, and how the community can use it.
So one thing I want to add is that can we work on AI tools or AI agents to help us clean data, harmonize data, centralize data, so that we don't have to put in too much labor?
Any question, any concern, thoughts from the audience?
So have in-depth discussions. If there's a quick clarifying question, just shout out loud. Otherwise, we move on to the next one.
So I guess we'll start. We had a also very lively discussion about data quality. Data scientists spend 90% of their time cleaning the data and doing data wrangling. So everyone among 12 people in our group had comments about their issues with data quality. And some examples included that type 2 diabetes, type 1 diabetes is often coded as type 1 diabetes, and you would think it's a mistake, but actually people do it for authorization, for example, GLP-1 pre-authorization purposes. So there are a lot of gotchas in the data which you cannot figure out without domain knowledge. Or for example, if you're trying to work with continuous glucose monitor data, but also overlap diet data recorded by humans, the data quality would be drastically different, which makes it so hard. And we talked about how it would be nice to use AI to automate these tasks, such as especially creation of metadata and data cleaning. And we had 3 takeaways, and 2 of them are probably what you expect, and the third one is a little less obvious. So the first one was to obviously develop and implement data quality metrics consistently. And there are some good examples of data sets which are of high quality, such as Kidney Precision Project and AI-READI dataset about which we heard today earlier. And the second takeaway was to develop metadata standards and incentivize using them, because often the incentive is almost reverse, spending a lot of time on metadata documentation is actually wouldn't make sense for a researcher. It would make sense maybe 10 years later for other researchers who are using the data. But our final conclusion for this topic was more related to equity. For example, for electronic health record data, they are of low quality, but it's very important to use this data to make the insights from data actionable. Therefore, we also believe that it's important to develop methodologies for using sparse, low quality, inconsistent quality data to promote equity and to be able to use data which, for example, come from rural areas or from minority patients. Thank you.
So we don't have a huge amount to add from what's been discussed by the other groups, but one point that did come up on both days across a number of topics was some of the redundancy that happens when people manage to get access to a data set, they clean it up for a analysis, but then they're not able to share their findings from that analysis, having put all that effort and person power and resources into generating data. So one of the things we were interested in was how to minimize redundancy with data QC and cleaning up. That was something new. Anything else for you?
Well, I think we're all scholars, so the methodology really works. I want to thank our note taker. We actually have note taker, we have discussions, we have the whiteboard, and then we use some AI to help us summarize and we proofread it. So I'm gonna actually read out 4 basic points that we discussed and maybe see if you agree. So we talk about primarily focus on integration related challenges. In data standardization, we talk about the importance of standards and labels. We talk about the importance of getting AI ready around the FAIR principles. We talk about the gaps in ontology that's lagging behind. In the data access pillar, we talk about the challenges in performing secondary analysis because they don't usually have access to the data. We felt that there might be new needs for new access models that facilitate the information exchange and think about risk-based models. For collaboration, we're going to leave it to the number 4 because that's a special topic. And we felt that the future directions of the data, there's technology component, there's a federated databases that we can actually make sure that people still own it and that we do it together. NIH need to play roles and support the whole system of standard access
And then engagement and training is very important.
So our group spoke about some additional issues. The biggest one that came up is data availability and having institutions willing to collaborate, share data, especially for electronic health record data and health systems. There also needs to be, I think a lot of the data sharing is required for NIH-funded studies. However, they also need to allocate sufficient budgets and time for making data sets available in a way that they're actually usable with the additional metadata and everything that has been described. It's a lot of time and resources to create a really good annotation. But with shorter and shorter grants, there's just less time available to make that possible. And then finally, I think often, especially for clinical data, it has to be de-identified to comply with standards and safety protocols, which precludes a lot of the linkages that then need to happen to do additional analyses.
Okay, great. So we're going to move to our next topic, which will be focused on the diabetes. So in this round, we're going to start from Orange Group. Okay, go ahead.
We talked about the importance of predictive modeling given the complexity and heterogeneity of diabetes. And something that participants brought up was lack of high quality AI-ready multimodal data sets, need for longitudinal data, and low quality of EHR data, even though they have high potential. And most of US healthcare systems now use electronic health records. Actually, almost every patient in the United States have their records in these systems. This data still cannot be used in hypothesis generative research effectively. We also talk about importance of integrating existing knowledge from guidelines and PubMed papers into such predictive modeling and how it's different given the lack of metadata in papers, and that AI could be used to promote integration of existing knowledge into this type of research. We also talked about integration of preclinical data, for example, using simulation studies to prepare for clinical studies. But I think the main insight was that there was so much progress in the field in the past 3 years that some researchers in the field, they don't even know about available methodologies as well as available datasets. For example, there was a talk today about AI-READI dataset, which is actually exactly the type of dataset, it seems, which participants felt that was lacking. So I think there is a need to address this gap in existing resources and existing modeling methodologies versus the knowledge which average researcher has.
So I'm fortunate I'm in the same argument with Polina. She's doing an amazing job. So I want to add to the diabetic perspective. There were quite a bit of discussion regarding a consistency in insulin dosing, how that would actually alter the data sets that used to treat these predictive models. And there's also quite a bit of discussion on missing values in both temporal longitudinal studies and what are the things we should do to impute or overcome to deal with the diabetic data collection for predictive models. Thanks.
Thank you, Dr. Peng.
So some of the areas that we discussed that I think haven't been touched upon yet are the sparsity of longitudinal data available for models, particularly from contemporary data sets. A lot of the data that we rely on is 20 to 30 years old from historic collections and may not perhaps best represent the current environment that we're in. There was also a discussion around concerns that a lot of what we were focusing on was extremely glucose-centric, and that it would be advantageous to be diversifying the phenotypes that we collect in the pre-diabetes state so that we could look at those in a longitudinal manner. I think wearables, we again had a long conversation around wearables, probably not surprising since we had Mike Snyder in our team, but I think that's been touched upon. And another area which we felt that was not currently explored to its highest potential was engagement with pharma and being able to work with data sets that they were generating in clinical trials.
Our group touched on a lot of the same things. Some others that were brought up is availability of environmental data and kind of contextual data, whether it's like GP location of the individual, things like obviously their credit card transactions, environmental data that all influence diabetes management and behaviors, but aren't generally available to researchers, whether because consumer data is either expensive, not available, or not permitted by IRB for linkages. And the other concept that came up is kind of broadening the definition and the perception of precision medicine, that it's really thinking about it as more than just genetic analysis or kind of omics data, but thinking about behavioral data, treatment plans, patient-reported outcomes and experiences as our ultimate goal is kind of outcomes that matter to people living with diabetes and improving their quality of life.
Okay, so we have one thing to add, which is the lack of balance of different modalities. Some are really rich, some are a little bit poor. So how do we balance this? How do we think about the contribution to our models? Will the model be overwhelmed by the rich domain, but not so good for the poor modality? So that's one thing.
Some of the key challenges that are discussed and which we would add, definitely, sometimes, the data is sparse. So sometimes, some of the information are missing. But one key thing that was brought up during our discussion, it is to model the heterogeneity in a data. Sometimes it's important to find out what is common, right? What is well conserved across different data sets or different domains, to model that invariant to understand data heterogeneity. And in order to get an AI-ready data set, the other thing that we discussed about, to make the data more open, right? That's one of the key discussion happened, that open source data sets could be useful for helping with model design. Yeah.
Okay, great summary. Now we can move out to topic 3, which focus on ethics. For this group, the blue group will be going to start first. Yes.
Well, I felt that we had a really great discussion. There's a national news, and I'd like to share with everyone. There's a news that you can find that the Grok just announced that they're accepting EHR data submission by users, and you can submit all the clinical tests and the imaging result to a private company, and hopefully they can achieve great precision and the great benefit. But we highly doubt that this is the way, or And then we believe that there are multiple issues that we need to think about, right? So I think maybe you.
Yeah.
Yeah.
So the transcript from our discussion that Elizabeth kindly put together for us is that our discussion really fell upon three major areas. We talked about ethical concerns. Major issues were bias, patient data security, and ensuring fairness. We debated the risks of transferring AI-trained models between institutions and the challenges of managing data privacy without restricting innovation. The second guiding question was around bias and fairness. We discussed how models could reduce biases but acknowledged the limitations in training data, especially for marginalized populations. Concerns were raised by our team over the over-reliance on models trained on homogeneous data, leading to increased inequities. The final area we spent time on was coalesced around privacy and innovation policies. We suggested the need for policies ensuring ethical AI use whilst simultaneously promoting innovation. We spent a lot of time discussing public mistrust and also thinking about the impact of social media in the area of data mistrust and mistrust of scientists and clinicians. And we also discussed advocating for a new ethical framework for AI. Our conversation concluded with a focus on collaboration and identifying ways that we could bring different stakeholders together to address some of these major challenges across an interdisciplinary focus.
It's very hard to follow Group Blue because it was a very thorough discussion. I think what we'd like to add to that from what we spoke about, I think was one, kind of building on the standards for ethical AI, also having standards and guidance for institutions to be able to implement them consistently and uniformly to kind of promote research, especially I think for institutions that serve disadvantaged populations where there's even more mistrust and the standard for data sharing and privacy is a lot higher. I think that's something that resonated for me from being from Maryland and Baltimore. We also spoke about the importance of engaging patients or people living with the conditions that we care about to really understand what is important to them and how they perceive fairness and ethical issues.
So we talked about the underrepresented group issue. And we also think that it's good to consider, if we go out to pursue this underrepresented group, to consider the burden of testing to them and how they perceive being tested. So that's one thing we talked about. So next thing is about the model vulnerability to attacks. So we talked about, are there safeguards inside the model or keys or private keys, public keys, all those kind of potential technologies to safeguard the models?
Nothing much to add. I think most of it's covered. I think one thing we also talked about, maybe to in addition, that can we bring some of the technologies already out there in other areas, for example, in defense and security, right, which Dr. Kui just mentioned. And the one last thing I would mention about the model explainability. So we touched upon the model explainability. So this is something possibly in imaging area or image analysis domain we talked about a lot. But in text data context or unstructured text data context where people are working on this domain of EHR data, question about explainability is still open and there must be lots to do. Yeah, so that's what, we had a discussion on that.
I want to add one last thing. One of our trainees mentioned that their required reading book is Weapon of Mass Destruction. And
Not mass destruction, math destruction.
I don't think we have too much to add from what our colleagues have brought forward from this discussion. I will mention just a couple of additional things that our group spoke about. One was the importance of local IRB groups and institutions in upholding some of the standards and the need to encourage a more uniform application of standards, some of which are already available but could perhaps be better communicated. We talked about patient and sample de-identification, the growing risks of re-identifying individuals or samples with AI approaches, and how to deal with that, whether that needs to be dealt with stricter application of de-identification protocols, or whether that's dealt with downstream, with the inevitable potential for re-identification. There should be some controls in place for usage and protection of that information if it does happen. We talked finally about really the need to involve ethics researchers in this conversation and experts in AI ethics in what we're doing.
Yeah, I would like to add that with the power of the models growing and the multimodality of data increasing, these re-identification concerns are also increasing. So that's why we spend so much time talking about ways to deal with it.
Oh, and separation of data and knowledge as one potential way forward.
Yeah, we talked about how to use knowledge graphs so that knowledge is actually stored separately from the data, and there could be more techniques which would help with these re-identification issues.
Okay, great. That's a great summary in here. I think the first 3 topics, we're kind of trying to focus on different angle in there, either on AI or diabetes or ethics. Now we move into our final topics, which is kind of trying to bring everything together. That's collaboration. I know this is kind of a very hot topic in there, so go ahead to start your summary. It will be purple group, start.
Hello, everyone. We have discussed quite a few interesting ideas for collaboration I want to share with everyone. One side, we started again from the dataset perspective. Right now, with all the open dataset, what else can we do to make it more enabling for collaboration? And there's ideas of maybe dataset alone is not enough. We have to have metadata, have the example, code base, how to analyze that data should be shared as well. And then I think our team also discussed about the potential organizing some kind of hackathon and data competition to really make those data sets being used by many. And so that's the one type of collaboration, concrete collaboration about data set we have discussed. And then the other thing, we talk about education. So this is a very interdisciplinary topics. I think we have biostatistician talk about her training on the biology, on the basic science side of things, to really have this common language to talk to biologists and clinicians to be effective. And also have someone shared about on this basic science side, they have to take some coding course to be able to understand how statistician, computer scientists work. Right? This type of interdisciplinary education could be useful in the long run. And I think finally, we also talked about organizing some workshop like this in other disease-specific conferences, right? Workshop is amazing. It's really truly interdisciplinary with computer scientists, domain experts, clinicians, biologists, right? System engineering, everyone. We felt this type of small scale workshop, AI workshop, organized in those conferences could be quite valuable. Anything you want to add?
Yeah, I think a lot was spoken about just bringing us together so that we develop trust relationships and learn from each other, and often take the tools from all of these different disciplines toward, to be able to answer problems that individually we could not solve. So in the kind of thinking back to the pre-COVID world when we were in physical proximity to each other, and how these collaborative institutes and centers are really important because again, they bring people together. And when that's not, and how can that be reproduced more artificially or during conferences, to be inviting people to like our conferences who have different interests and different skill sets, and to broaden that exposure to each other. And finally, making sure that when we think about collaboration, it's not just, just clinicians, basic scientists, biologists, but also thinking about people who are living with a disease, and they are part of our team to help make sure that our research that we do ultimately addresses a need that is important to them.
We focused on the trainees quite a bit in the discussion. So one thing our trainees expressed was that to be trained in a multidisciplinary environment is critical for this type of research. So NIH can encourage or initiate some training programs that are multidisciplinary and set up the environment for them. It would be great. And the second thing is that for some immediate collaborations trainees face, like it would be great to get some quick knowledge, like a 15-minute YouTube video, high quality on various topics, well organized by experts in various fields. And that would be great to facilitate not just our trainees, but also everybody in the field. It would be great if NIH organized it.
Thank you. I think most of it is covered by you and Dr. Kui. And one thing we came up, right, definitely developing a course curriculum in a university level, there are lots of barriers there. So this type of YouTube videos or NIH initiative, like now what we are doing now, these are very useful to teach or train trainees or our next generation on the understanding collaboration. Also, it was discussed developing a new set of curriculum where things will be integrated, not like, two different departments will work, but integrated curriculum. It's very, very difficult. That does not exist. And one solution is YouTube video, but for university context, it needs like new courseworks and new way of, adding value, like how the trainees are seeing, like how they are valued, right, for doing this kind of curriculum. The other thing we discussed about, like which is already there possibly, like more investment or more reward for multidisciplinary team science, right? One cannot work in silos, so different groups need to work together. So when someone is writing grant or, grant evaluation criteria, maybe specifics needs to be made there, multidisciplinary team science approach would get more points or something like that reward. So we discussed that the need for more and more, right, clinicians, practicing scientists in clinic and computer engineers to work together in upcoming R1s or any research grant submission. Thank you.
I think our discussion echoed much of what you've heard already, with the key theme being the need for improved communication and training to be able to speak the language that crosses these different silos. We discussed some, potential mechanisms for that kind of training, whether it be through increased numbers of slots for training awards through current mechanisms, or whether there are other programs that might facilitate pilot projects and things like that. We talked a lot about the depth of knowledge that this cross-disciplinary training might require. Does a trainee need to be completely fluent in both sides of these equations, or is bringing a biomedical trainee, for example, in for some cursory training in computer science and machine learning appropriate? And I think where we really came to was that it probably is person and project and team dependent. I think what was clear, though, is that working in the silos, for example, having computer scientists give a model to a biomedical researcher and having them go crazy with it completely separately, probably isn't the way to go. We talked a lot as well about trust, and what is the point of all this? And that is building trust in a model, in the results, in the data. We talked about the need for validation cohorts or validation data, replication data sets, and things like that. So some of the specifics maybe to how these kinds of collaborations would work. I don't know if you have more to add.
And we also took a vote at the end in terms of collaboration versus autonomous AI tools, and 9 people out of 11 voted for collaboration, with 2 people saying that actually it would be really nice to have more automated AI pathways, especially with language agents available now. Maybe AI can help to train biomedical scientists in the use of AI.
All right, so we're the last group to actually report, but I felt that we have agreed with it, all have been said, but with some additional messages. First, I don't know whether it was intentional by design or not, but we felt that for Anna from Stanford and me from UAB and students, we got people from industry, from world famous research lab, and then from upcoming research labs. By itself, we come to this breakout session feeling a little uneasy and think, are you competing with me or what? But then the end result is we actually collaborated together in real time, come up with really good guidelines. And that showcased that we are one of the good examples, maybe other group as well, that we have the soft skills, and having soft skills, communicating respect is very essential to overcome the barriers of culture communications. From the technology point of view, we felt that having some portal standards together, and that is very important baseline. But collaboration is not just about accessing data. It's also about learning and become more actively engaged across discipline. You have to learn something beyond computer science if you're a data scientist, and then learn some diabetes so those are the comments. And we also pick out our favorite, my favorite project, like a Bridge to AI as an example. In Bridge AI, we produce data set, but then people and ethics, these 3 pillars define the Bridge to AI. So the people is all about collaboration. We were thrown together from 1,000 investigators all over the nation. And now I think that you've seen some examples and their report, how wonderful data set there is. We also brought up the issue of collaboration among senior people, especially how do we sustain the collaboration? And then there may be new creative ways to evaluate it that goes beyond having a paper. And then maybe do you have a reputation of building successful community? And then that could be building to the future evaluation form. Thank you.
Okay, that's really great summary for all the topics. Let's give another applause for all the co-leaders with all these summaries. I want to emphasize, I know a lot of people trying to take notice. So all our meeting already been recorded. We're going to do the summary on our website. So just pay attention to our workshop website, which will be all the seminars, recordings will be put in there. Now we'll have a few minutes break until 1:45 to start our final panelist discussion. So hopefully everybody will come back to 1:45. We'll hope finish everything on time.
Sorry, let me add something. I would encourage everyone to come back maybe a couple minutes before 1:45. Make sure we start on time so we can let people go to catch your flights. Yeah, thank you. And yeah, we'll be loading the slides for panelists. If you all would not mind, maybe come up to check, make sure your slides are loaded fine. Thank you.
Yeah. We just started. No, sorry. Yeah, it's 5 years.
I borrowed the picture. Yeah, that's nice.
Yeah. Just remember, we're going to let Mike go first.
Yes, let Mike go. Is his slides before my slides?
I'm loading it right now.
Oh, okay. Yeah.
Yeah.
What time?
Yeah,
that's loading. Yeah, they have their own slide. Are they loaded? Yeah, right now.
Loading right now. Yeah.
All right, I'll sit on the end. Are you sitting there? Standing up 2%.
Sit down. Either way. If you maybe sit down, we can help advance your slides to make it easy. You can just we'll leave you guys. Actually, you can. Okay. Well, we can try to control the slides.
Okay.
It's here. Okay. He sent us. He used this one now. This is the long one. So how? Let me see here, right?
Yeah, you just double click that. Double click.
Why it's so long?
Yeah, there's so we copy everything.
Yeah, very like he said, you know, she like you.
Okay.
So.
Only imagine efficient. Yeah, I saved the 10 minutes. That's great.
So this will be go first, and then
Wei,
and then Jack.
Yeah.
Jeff is.
I named the day.
Oh, okay. Data ecosystem.
No, that's not. They have like a lot of time. It's a really nice dance, like, yeah, it's like a blend. It's like, I'm sure like soon there's gonna be a map. You know, like I think this masters of data science kind of new within the last 5 years. It's programs. So really, I mean, I don't know what's like get to that AI state. You know, we're talking about breakouts and collaboration. So I mean, I guess it's like you get that knowledge through hands-on work maybe, and there's like a bunch of training. No, there's no like formal disciplines. You know what I mean? Maybe if you go the PhD route and you go where there's something.
That's right, yes, I have seen.
Yeah.
I mean, originally.
Yeah. Yeah. A lot of these people start out software. There's like.
Everything's loading here. So my constraint will be first one, Wei is second one, and Jeffrey third.
Yes.
And-
Oh, okay. Actually, can you make it a different order? Sorry.
Okay, yes.
Maybe because, well,
Yeah.
he said he has to leave a little early.
Yes.
Maybe he's first.
Yeah, he's first. Yeah.
And
Let's
then.
say that will be first, and then
Okay,
Wei
after
is
that,
second,
yeah.
and then Jeffrey third.
Okay.
Dr. Washington is the last.
Yeah.
Oh, okay, yeah, so I see the point. Yeah.
Okay, so what, you know, I think taking first course, you I'm sure.
Of this size. As the next closing.
Oh, okay.
Yeah, okay.
So let's go to the top.
Nice.
Yes,
That's a.
What is that? Will you help? No, you, no, how do I do it?
So you just click either this one or.
Oh, yes,
this
that
one.
part, but how do I share?
Right now it's already shown, so.
Oh,
if you
somebody
show-
else
Yeah,
share?
if you.
I'm sorry,
Just-
oh, that one, yes. This,
In case kind of have anything problem, I will see. besides
okay,
the-
thank you.
So this one you just click share and then.
Yeah.
You want to check your slides?
Need to apologize. I need to leave in the middle of a session to catch a flight.
Oh,
It's very nice to meet you. We'll follow up. Nice to meet you.
a lot of, some of the speakers just want to sit.
Yeah,
Here, but there is
yeah, yeah, yeah.
So you can just tell us when to,
Okay, that's fine.
move.
So you do. I
You
said
can
like.
crossword.
Yeah, so this one.
It's like,
You need a.
Yeah, yep, okay.
very nice, and I learned.
Yeah, you get to remind me.
Other. Modern.
We want to finish a little early.
Yes, hopefully we get on time.
Okay, okay.
Probably maybe early. So this is minutes in case.
Okay.
Yeah. I usually.
Well, there are going to be more questions.
Yeah.
So I think we'll just stop the questions.
Find.
Is that what we're doing?
Yes. Yeah, everything's ready. So.
Like running it from.
Thursday?
My ID.
Keeping that.
Command line. Yeah.
Okay.
Yes. Time with your family, friends. Like, yeah, well, interesting. I've been memorable about going to the kind of.
I'll find out. Oh, yeah.
You know, every day.
Like 4 times, 5 times.
Yeah. No, I think it's good.
Yeah, well.
Like, oh,
So I think my first couple of slides all cover what was presented before. So
That's right. So I thought you were. Yeah.
Yeah, we're just
That's awesome. Yeah. Okay. I know, right?
Yeah, I think it's all uploaded. Do they want to advance from the seeds? They could try that thing.
Thank you very much.
We do that right after undergrad or.
Yeah.
That's true.
Thank you.
more efficient.
That'd be great.
I see.
Yeah, we'll wait.
Got a guy walking.
Yeah, maybe I will start, introduce myself, and then you'll do next.
No, I literally just downloaded it.
Yes, first
So
of all.
we don't have to fill out.
Yeah, perfect.
What I should have done was built an AI.
Oh, there you go. Or I was, yeah, send it to Notebook LM, like for the podcast.
Yeah? I haven't tried that.
It actually.
Okay, should we get going?
Scary.
We need the.
You do this. Okay, good afternoon, everyone. Let's start our last session. And first, thank you for staying here until the last session. And just introduction of myself. I'm Chen. I'm a professor from Weill Cornell Medicine. I'm also the multiple PI for dkNET and PanKbase.
Hello, I'm Katerina Kekris, Professor of Biostatistics and Informatics at the Colorado School of Public Health. And we'll be moderating together.
Yeah.
This session will be a little different. We're going to have quick introductions for each of our panelists, and then they'll lead a summary of their topic, and then we'll take questions. So I'll get going. Our first panelist is Dr. Michael Snyder, the Stanford Asherman Professor and Chair of Genetics at Stanford University and the Director of the Center of Genomics and Personalized Medicine. He is a leader in the field of functional genomics and multiomics and a major contributor to the ENCODE project. Dr. Snyder has initiated the big data approach to healthcare through his work using omics to detect early stage disease, including wearables to detect infectious diseases like COVID-19 and at-home microsampling to measure hundreds of molecules from a single drop of blood. He is the first researcher to gather petabytes of data on individuals and has, and in addition to starting more than a dozen companies. Then our next panelist is Dr. Talitha Washington, Professor of Mathematics at Clark Atlanta University. She has research interests in applied mathematics, dynamical systems, data science, AI, and education. Dr. Washington has received several prestigious awards, including the NSF Women's History Maker Award, the Howard University Outstanding Faculty Award, and Fellow of the American Mathematical Society. She is a fellow and also president of the Association for Women in Mathematics. Dr. Washington is leading important data science initiatives, including directing the NSF National Data Science Alliance and the Data Science Initiative of the Atlanta University Center Consortium, focused on expanding data science research and curriculum across historically Black colleges and universities.
Yeah, and I will introduce the next two panelists. Dr. Weizmann is a Linda Crondall Professor in Computer Science and Computer Medicine at UCLA. And she is a leading expert in AI data science and their application in biomedical fields. And she is also a multiple PI and also lead PI in like dkNET and Bridge AI and some other program. I won't go through all of them. So another panelist joining us today is Dr. Jeff Gregory. He is Associate Director of the Center for Research in Biological System at UCSD School of Medicine. And he is a PI of NIDDK Informative Network, dkNET. And he has more than 2 decades experience in providing informatics environment for biomedical research to enhance the scientific rigor and the reproducibilities. And both Dr. Wang and Dr. Gregory, they are the co-chair and organizer for this workshop. So yeah. We'll start.
All right, thanks. Well, I'm Mike Snyder, as you just heard. And my topic is challenges and opportunities in type 2 diabetes. So what I did was I assembled what I thought I heard through the last 2 days and they're put up there as bullet points, and I will editorialize on top of that. So obviously we're in an incredible era when you think about there's new compute tools and approaches. And I think quite frankly, we only touched on a very, very small fraction of what's out there. When you think about all the different tools that people can use, there's a lot of whole imaging kinds of models people are using to represent data that I haven't seen come up. So I think there's incredible opportunities there. I think there's incredible opportunities, not just with all the new technologies, I've listed a few up there, but there are many other data, collecting data. There's many, many ways to collect data now that are very creative and many more to come, I'm sure. And you heard about some of those at the meeting. So what we want to do obviously is marry these and you know, they will come together. AI is important. I'm sure all of you use it. It's literally in every aspect of everything our lab does these days. I can't think of a single thing we do that doesn't have some form of AI in it. And so, as I say, I've assembled some of these bullets here that I heard throughout the meeting. I think the first 3 slides, actually, we just heard a lot about from the previous panel. So I'll go over these super quick. You know, getting in representative data. I think one thing I haven't heard, how do you know when you have got enough data for a good model? What are the QC? I know that was touched on, but I think getting proper QC, so we know we've really covered and actually increased sampling. So we know we've really got what we want. Not that you ever have enough. And another related question is there such a thing as too much data? And in my mind, the answer is no, but not everyone agrees with that. I think getting data in a form that, you know, everybody can use, we all agree with that. And I think I thought it was refreshing this morning to see all the big projects going on, people standardizing data and things. Hate to say it, but every consortium I've been is doing the exact same thing. And somehow they've got to get married across these consortia and they all reinvent the same standards of pipelines, which are almost matching, but never quite right. So I think NIH should take the bull by the horns, in my opinion, and really get the standardized across all institutes and things. I know there's a common fund project to do that. Sharing data, being my bonnet, I still can't understand why CGM data is not out there. It's almost impossible to get. And the researchers always blame it on privacy, but the reality is patients are happy to share their data. It's usually the researchers who don't. Let's call, you know, call it what it is. So I think we need to really push that harder. I think you should get rewarded when you share data in some fashion. I think how to build that in, I think it is possible to do. Yeah, we need many, many more tools. All right. So I wanted to dig in this a little bit deeper. Diabetes is really super heterogeneous. It involves many organ systems, liver, pancreas. You know, your brain is your number one energy or one of your top energy consumers, right? Muscle, obviously. And obviously there's a ton of different biochemical pathways that are all involved as well. And so when you think about it, it's not surprising that we are, you know, a balanced homeostatic system across our organs and across our microbiome and such. So I think understanding how that all plays and then what perturbations actually set things off balance is really what diabetes is all about. So at the end of the day, you know, how many subtypes are there? Well, it's probably continuing and people will have multiple subtypes that we heard about genetics here. We now have, here's a good AI one. We now have new AI methods for analyzing genomes. This idea about setting up linear polygenic risk scores, I think is very outdated. It doesn't work that well in spite of people saying it works for the top couple percent, maybe 2 to 4%. Doesn't work well for the bulk of people. So I think we don't really have the genetic basis of most complex disease. And I think these new AI methods, you can take a look at some of our papers up there. We have one on diabetes now on bioRxiv. You can take a look at. I think they're just finding 10 to 100 times more genes than about previously and a lot more of the heritability. I think you can also define diabetes mechanistically, like insulin resistance, muscle insulin resistance, liver insulin resistance, beta cell defects, et cetera. You can also define it molecularly and microbially for some of this today. And clinically, I think at the end, people say, well, it's only the clinical part that matters. I don't agree with that. I think we need a better understanding of the first part because the two feed each other. Meaning I'm on GLP-1 and I don't want to be because I get nausea and I don't like that. So I think we want to really have lots of options that are very specifically tailored to the subtype you are. I think that is a very doable goal. And yeah, so I think it'll be a cycle where as we understand more, we'll build better treatments and go back and understand the subtypes better. All right. Forgot to say, one other thing on data sharing, which is there's a huge bottleneck around this privacy and cumbersome, especially at the university level. You should be able to push one button and get data, not do 9 months of back and forth DUAs and all that stuff. And if somebody could have a generic form that everybody could agree to, we'd save millions of dollars. We'd spend it on more useful things and lots of headaches. I just find that terrible. All right. Ultimately, why do we want all this data? We want to build predictive models. We want to know who's going to get diabetes from the time they're born, maybe before they're born, because I think the maternal effect's probably there. We need to predict progression. We need to predict optimal treatment. And I think one thing we haven't emphasized this so much, but longitudinal data is going to be super powerful for everything we do. So I think collecting that kind of data will be useful. And then I think we should be able to use AI, maybe that'll get talked about, for treating diabetes as well, designing new drugs and such. This might have been covered already. Do we have the right workforce? I think so, but I think we can build more hybrid workers. I think, used to be bioinformatics people were very separate from data collectors. Now they're often indistinguishable. I think that'll be true for machine learners, deep learners, et cetera, and folks as well. We'll skip over that. Here's where I think we can do a lot better. We're just hitting the tip of the iceberg. I think Eran gave a great talk on this. Our group's very engaged in this. We are multifactorial, and we need to understand, food is one of the number one things we do, especially impacting diabetes, exercise, sleep, and the timing of all this. When should you exercise? Well, anytime you exercise is good, but the party line says exercise in the afternoon. Well, that's true if you're insulin sensitive. We have a study that we've got submitted, but it's not true. It's on biomarker you see. But if you're insulin resistant, you should exercise in the morning. You'll get better glucose control the next day. Timing is a big deal. We don't incorporate that anywhere. So everything on that board from the first 3 bullets should also have the timing aspect as part of it too. And you heard about Eran talking about building integrative lifestyle models. I think we could do this with incredible precision. Again, because we all have our genetic features, our molecular features, our mechanistic features, and then we bring in all the external features that's listed there, bullets 1 through 4. And I think we can build fairly precise predictive models. And I know to some extent I have this on me. I know exactly how much, if I ate 2 pieces of pizza, I put myself to sleep. So there's you can build these kinds of models at a personal level. And I think ultimately that's where all this is going to go. I think I may have spent more than 4 minutes. So, okay, over to you.
Okay, so my assigned task is challenges and opportunities in AI. So I think Mike actually set really a great, very solid foundation for the discussion. So I'm going to quickly go over. Can you guys hear me? Yeah, I'm going to quickly go over a few things that actually I learned from this workshop and that I feel that are very important for this community. The first is, as Mike already mentioned, the data heterogeneity and the disease heterogeneity and also data heterogeneity, because diabetes is very heterogeneous. As a result, when people study this, and there's a data, actually a lot of effort over the time, and then collect the data, right? So each project actually they maybe have their own focus, so that they may collect a few data modalities, and then for a different team they collect somewhat different data modalities. So you can see, if you can imagine this as somehow the the data modality versus the tasks, right? So this matrix may not be fully complete, right? There's many empty parts. And then traditionally in machine learning field or in other field, analytics field, we tend to sort of like either impute the data or we just drop the ones with incomplete data. But in our case, we try to integrate across very diverse data source, right? This becomes not an option anymore, right? Because either you end up with predicting or imputing a lot of data that did not exist, or you end up dropping a lot of data, right? So that's require actually the AI and machine learning model to be mindful of this type of data heterogeneity, right? So be able to leverage all the available data, but not try to inject in a lot of artificial signals that where, you didn't really collect the data, right? So don't have to go through the data imputation step. Actually, has been quite a lot of effort especially with the help of foundation model. Actually, people are developing model that fuse different type of data. This is something that that's one research area. And also I mentioned this multimodality and also nowadays, like we have heard actually even in these meetings, right? There's many presentations touched upon large language model, foundation model that has already been developed on different data set, right? So, and then actually, if you look at this figure, very busy figure that actually I steal from one of our own papers that's to be published in a couple of weeks, is we catalogs all the currently, actually this is still a partial list of developed foundation model. Was it their type? Was there fundamental technology? Was a difference there? But if you look at our paper, there are already quite a lot of different foundation model being built, and then how do we use them, right? So I don't think it's actually cost effective. Is everybody try to train their own, right? One could be because not everybody have the resources like Microsoft or OpenAI has. So, and the second would be like actually it's we all have different data, right? So sometimes when data has like privacy concerns, like we cannot directly share data, but how about sharing the model that to be trained, right? So if we train our model in a proper way so that we can safely guard the privacy of the patient that who contribute the data, then we can share the model, right? In a more open way than we share the data, right? So, and also once people like me as a AI researcher, we get access to all the models, actually, we could possibly like build some kind of a meta model, right? Or try to leverage each of these models, like maybe treat them as a special agent, like mixture of expert model right now. So it's like we can, assuming that each model has its own specialty and advantage, maybe is doing very well in certain tasks, but not for all the tasks, right? So then we can leverage the strengths of each model and try to integrate, build a meta model of that. So that is something that we could think of. And we have also talked a lot about data privacy and ethics, right? So this is definitely important related to data sharing, model sharing, and also use policy. And even in the breakout group, we touch about this. Okay, maybe there should be also even regulations, policies, and laws, or because now AI become more and more powerful, which means in addition to like it's bring to a benefit to community, but also it may create some harm, right? If we do not use it properly. So there should be like a stronger regulation there. And also how do we engage the community and establish public trust? So, and then to do all of this, actually, one important fundamental aspect is data standardization and metadata standardization governance, right? Because even if we want to build AI model, right? So we talk about something like AI readiness. AI readiness has many different aspects, but having standardized data and metadata will be one of those aspects. And then we have also touched upon quite a lot on the data quality and the bias, right? So data bias may contribute to actually the model bias, and that's come to the question whether we can trust the AI model, right? So there's reliability issues, and also we also talk about like some of the presentation talk about hallucination, right? So this is one of the well-known problem and that the large foundation model like ChatGPT face, right? So, but there are ways to conquer that already, but not completely remove it, but at least reduce it to a great extent. And then something I want to touch upon, like encourage people to in this panel to talk is about human-centered AI, right? So that's involved many aspects. I only list a subset is like explainability of the model, right? After all, we want AI to serve human, right? Not the other way around. And also, how do we treat AI as agent to help to assist us, like the scientist, to really ask the scientific questions and then to treat the disease, have a better understanding of the disease and treatment, and making prediction about maybe the prognosis of the patient. And then last but not least, I want to mention this AI readiness. We have talked about a lot about this, not in this community, just in this community, but everywhere, right? So So across the different sectors of NIH, this AI readiness. So obviously in each of them may have our own definition, right? But there's multiple aspects of it. And then how to, what it is, how do we make more data AI ready? And then, so I don't have time, but I know I work with Dr. Pepe Ping, who's, have done a lot of work in this space lately. So I would encourage, we have some discussion offline on that. So the last point I want to make is that we can actually also leverage AI to make data AI ready. So making data AI ready doesn't mean that each of us have to be put in more manual work, right? So that to cure the data manually and then to do things manually, but we should be able to leverage AI in every step of this process to make it more automated, to make it more consistent, more AI ready.
Okay, so my task was also to focus on the data, and actually the prior two summaries, I mean, a lot of it was directed at the data, right? And so, we've been talking a lot about data over the last 2 days, and there is a growing data ecosystem, where we have, a number of, NIDDK funded resources, heard from the central repository where there's a lot of data, a PanKbase, which is built off things like PanKdB, Radiant, we've heard about All of Us, which is, a very large project. We have Bridge to AI, where we heard about AI-READI. There's also MotorPack, which is, a big set of data, that's just being released. And then, the Human Phenotype Project. So again, we have this very large growing data ecosystem, but is it ready, again, to be used, in the development of AI methods? And, I think, as we've been discussing, there's work that has to be done, to achieve that. And so I think, there are a number of sort of data challenges. And again, I think, we're sort of echoing one another, but, these are themes that have come through strongly. The need for large, diverse data sets, as Wei had mentioned, having the appropriate data, to develop these, the models. The data harmonization and standardization, again, is key. And, the issue, I think, is, we have sort of multiple steps in that process, right? So you have people who are collecting the data for their project, making that available, through, resources. But a lot of times, their processes are designed around sort of the hypotheses they have developed, right? Or in terms of the goals they have initially developed. And how do we then, work with groups to enable sort of this additional potential lift to make the data, AI ready? And Wei mentioned sort of the agents to assist with data harmonization. Again, if these agents are easy enough to use, right? Can we push them down even to, individual research groups to help with preparing to make their data fair, right? I mean, with NIH's new data management sharing policy, every research group now has to, make their data available, within the time frame of the award or upon publication. But the critical thing is that metadata generation, that harmonization upfront, right? The more we can, make it easier for researchers as they're collecting data to do this harmonization, I think the better off, everyone will be. And I think the other issue is sort of the recycling of that data. In terms of once one does data harmonization, where does that data go? So if you go into a, protected data space, you do your work, you publish your results, but, where does that data that you use, that you had, spent a lot of time, standardizing, harmonizing, a lot of times that might just be lost, right? And so how do we sort of build upon each other's efforts, so that it's a sort of a collaborative process? The access issues for human data, and there's a huge, diversity of ways to access data. AI-READI is a very simple data usage agreement, right? So a click-through process that that was mentioned earlier. Where other places, it's a much longer process, months upon months, local IRBs and things like that. For broader use with, AI, ML techniques, how can we sort of build upon, the knowledge, sort of the prior work in terms of consents to sort of start working and dealing with, pushing those forward. So, that we can have more open data for, some of these computer science problems, in terms of using that data. And that gets to, again, data governance, appropriate licensing, as this data is passed through into development of models, what does that entail in terms of, how a model can be used or shared? Because it's all starts with the data in terms of, the licensing on the data and how that model can be used. And, as was mentioned in some of the breakouts, the issue of using protected data within foundation models, and pushing that forward. So, AI-READI was mentioned, and 6 days ago, I think it is, the paper from Bridge to AI came out in bioRxiv. And again, I think this gets to an educational point as well. What is AI-READI? I talked to many people, and no one quite knows, what AI-READI means for their data, right? And so I think this is something, where when people are preparing their data, sharing their data, what are those standards? What is the required metadata? Issues around provenance. So there's a whole host of attributes that are required. And I think just like in the sort of data publishing sphere, the FAIR standards or the FAIR itself is not a prescriptive definition of what is needed in each field. FAIR is sort of a set of guiding principles. And then, as fields are working through, as repositories have been working through, making data publish, they have to specify the metadata and standards that are used. And I think, with AI-READI, it's going to be a similar exercise, as we push, those principles down, in terms of making the data available. And then, the last one, I had a quote that I heard, and I forget who mentioned it, right? But everyone wants to do the model work, not the data work, right? Again, the work of documenting data, curating data, and we've heard this over and over and over again, is very difficult. And so again, I think this, building on others, the reuse of well-curated data is going to be important because, to do the appropriate QC and all that is just an immense amount of work. And data scientists do this over and over and over again, and for similar and similar data sets. So how do we get out of that, process, and how do we make that more efficient? And I think that is, a key aspect of this whole data ecosystem, right? Is how do we build upon others' data and make sure that, we aren't redoing work that others have already done. So that's my last one, and then that's this.
All right, so I'm going to talk about workforce development and then also my thoughts. So yesterday when I was walking back to the hotel, I was talking with a colleague, and she was saying that her colleague was doing a social determinants of health study and basically kind of copied and pasted a survey collection method over. And in that survey, they looked at wine consumption. The original survey was for white women, but he was looking at Black men. And she told him, wine isn't, I mean, some Black men do drink wine, but more beer, more liquor, right? Why are you doing wine? You have to understand the population to collect the appropriate data if you're really going to look at social determinants of health. There has to be a cultural, societal understanding of the population. And so as I sit here the past couple of days, When I think about workforce, I think about workforce and those who are doing the research, because I kept hearing we have incomplete data sets. We don't have data sets for certain groups that are, I'll say, underrepresented in our data sets, and we have a hard time getting the data. Well, I don't know, I'll say this ambiguous. We have said that in order to collect data from the groups that are underrepresented, those groups need to be a part of the research design. Those groups need to be a part of the ones doing the research. If we have homogeneous research groups, then we enter in what's called groupthink, and then we have incomplete data, and then we get bias, we start to hallucinate, and what sort of remedies are we really proposing that makes for a healthier nation? And so these are the thoughts that go through my mind. So part of my work running these two centers is to really think about what are different ways that we can push for more from historically Black colleges and universities, from other MSIs as well, to do this thing of data, AI, ML, all the L, cyber, all the rest of that. And you'll see in the upper left, that's our end of year celebration where we celebrate our students who graduate. And on the lower right, that is it's a 2-week research intensive workshop for HBC faculty to dive into data and AI projects, some of which focused on health, to really think about ways in which from students to faculty, how can we get more participation in there? I'll be frank, I would like to see more HSI, HBCU, TCUP, and so on representatives here in the room with me. I'm not seeing it. And I think that's a part of our struggle, is that we really need to think about, I think, data collection, surveys, research, interpretation, model development with cultural nuances in mind, if we're really going to think about advancing health in a stewarded way, and one that's responsible. And it's not going to look the same as what we've done it. It may look different. And then I'm also work with Dr. Herman Taylor, who's at Morehouse School of Medicine. We work with the NIH project Aim Ahead, which really looks to bring together researchers from minority serving institutions, those in the community, those who are practitioners, to think about how can we create ecosystems to advance AI ML to bring forth health solutions in this collaborative way. Because in order to really advance it, there has to be, I'll say, an intersection and a union, perhaps, of all of these different fields to think about this, to bring it forward. And the way in which we think about workforce development, it's from, you'll see the lower left, from K-12, getting folks interested in robotics, which, you know, it's like self-driving cars, right? Through our pre-freshman experience, which is a 1-week program, you'll see there a badge where we looked at health justice, to bringing together faculty to talk about the data behind Black health and be very explicit about what does that mean, what needs to be done, how should it be done, and having those conversations. And then also we have a seminar series that was one of our more popular ones, where Avery talked about how he created it was dermatology solutions because his wife had passed of skin cancer, and the technology wasn't there for a person with her melanin. And so our work really thinks about how can we cultivate the workforce? How can we really think about doing this in a way that's slightly different? Because again, I'll just say that, so we don't have these blanket, with the round peg, square hole thing, you know, with our data collection methods, our model development, this is what should be, and this are the health solutions. I don't know. For me, it gives me, for lack of a better word, the EBGBs. I don't know if that's, it's not a technical term, but it makes me pause and then think, what can we do in order to make sure that we as a community don't have groupthink, that make sure we as a community really take into account the cultural nuances and the multi-layers in this country. And, you know, Tuesday's coming, we'll see what happens, right? But going forward in a way that's thoughtful, going forward in a way that really thinks about ways thinks about data collection, data processing, modeling, AI, all of these things that we're hearing today in a way that really responds well to different communities and also shows up with a respect for the different communities that we propose solutions, health solutions for.
Yeah, thank you. Thank all panelists for very nice introduction. And we forgot to mention from the beginning, this panel will be also monitored by Dr. Fan Feng from Vanderbilt to help about online questions. Thank you, Fan. So now the floor is open for questions. So if you have a question, please line up to the microphone. Yep. While we're waiting people to stand up, maybe I can start from a question for Michael. I just want to push a little bit on the subtyping. You mentioned that people have different data modality to do the subtyping, right? I'm just wondering, how do you feel we should do subtyping based on each modality and integrate it later, or integrate it now and then do subtyping based on the multimodality data?
I think there's value in going both directions. One is we don't understand in many cases a mechanistic basis of the subtype, like insulin resistance, things like this. Sometimes we do, sometimes we don't. And so I think we should get them, and we should actually, by the way, turn these into very simple tests. I think we're now capable of doing this. We've been doing a lot of subtyping for, just what I said, insulin resistance, muscle insulin resistance, some of these other things. We can now, with 0.9 confidence using AI, from just a simple at-home CGM test, drink a glucose shot, and we can actually tell with pretty high confidence your insulin resistance. So suddenly it's a very, very simple home test that costs $50 instead of thousands of dollars for the gold standard one we run. So I think we can bring those in. I think that will help us understand. It turns out most people are not, a lot have one dominant subtype, but they'll have several subtypes as well. So I think we're going to understand that, and then how they react to, here's where understanding subtypes, you can relate to food. It turns out if you're insulin resistant, you will spike in potatoes and pasta. If you're insulin sensitive, or resistant, you'll spike the rice, the same, pretty close to the same. So we can start relating lifestyle information with subtypes. And then I think we're not there yet, with building more predictive models, we could do even better about exactly what you should do when and for how long. And a simple example is if you're going to eat that cookie you ate at your lunch, you'll probably have to walk 25 minutes to get your glucose spike down to normal, right? If you had the fruit, well, maybe it's a 5 or 10-minute walk. So we can get very, very precise, I think, in our predictive models at an individual level. And I think you get there by going both directions. I don't know, that was a long answer, but I hope you got the gist.
Thank you.
Yeah, so. Well, thank you so much. I'm Chen from Harvard Medical College in Nashville, Tennessee. This is HBCU Medical College. This is a great 2 days session, and this is also my first time to be here. And Dr. Washington, I appreciate what you mentioned, like on the workforce department. I think it's important. This is what also we kind of talk about this when we look at the room. There's almost no Black students or Black researchers here. And that's what we are doing now. We have data science program, biomedical data science program, and all of 80% of our students are Black. So hopefully next year we're going to have more. I'll bring my student here, and we can make it more diverse. Thank you.
Yeah, I just talked to your colleague, Dr. Sajid Hussain. So Fisk and Meharry is one of our partners in this work. Meharry also runs a hub for the NIH AIM-Ahead as well. And your colleague, Dr. Sajid Hussain, is also a co-PI with me on our NSF National Data Science Alliance. So there's more work to be done and a lot more, I think, I agree with you, connections to be made.
Right. So at the same time, I also think it's important for the student, like consistent to have a degree, has really good foundation on the machine learning data science training.
So while we wait for others, I'll ask a question. And some form of this may have been already introduced during the workshop, but I wanted to bring it back again about the resource intensiveness of training AI. And that many, it may not be accessible for institutions that don't have the infrastructure and the financial sort of ability to fund something at that level. There's a movement called AI democratization to make it more accessible to a wider range of people and organizations. Can you comment on that at your institution, or have you seen any applications of this? Oh, yes, it's just about making AI, since it's so resource intensive, it's not accessible to many institutions and organizations. How can we make it more accessible if you've seen anything from your perspective?
Let me give a first try. So I think this is question I could apply to everyone. For AI education, I think that there's a multiple aspect, right? So for me, from the AI side, actually we have been doing a lot of outreach activities in educating, a broader community about the basic AI concept, what to do, right? We have a, training program not only for the, undergraduate student from other disciplines, especially from underrepresented community, but also all the way to the high school student. Actually, I know Chen just left, so he need to leave early, but he and I organize a program called Dataset for All. It's for the high school program every summer. So at UCLA, we also have this, they call the Breakthrough Tech AI program. Originally started from Cornell, but now they have 3 campuses, Cornell, MIT, and UCLA. So I teach that for the UCLA side. So every summer, it's a full summer session, 10 weeks. It's this past summer have more than 200 students enrolled. So we teach them the basic AI, how they get their very first project started, and then we even line them up with their very first internship. So with company in this case, because we have a lot of company buy in this. For the computing resources, actually, we are also facing that challenge, right? So doing AI, it's very expensive. And then you can see that's why the major players you can see, for building a foundation model are those major companies, right? So for academia, we do have some local computing resources, and then but there's cloud resources which not cheap either. I think from what I know, there's relatively new resources. It's called NIRR, NIRR, right? Everyone can apply for the cloud credit. So you can, request like, AWS, OpenAI, or from large supercomputing centers. So you can submit the proposal to request that. Well, you can't request a lot. So for us, like we request like 50K and then for a year. So, and then that's helps to some extent. So I would encourage everyone to, to submit that. I think they have the rolling basis for submission. That's what I know. And then also I heard like DOE also have a large facilities that they, if you have a collaboration with DOE labs, that's also another possibility. And another thing is that for large foundation model, it's like there's a lot of model already been built, right? So now the question is, like I just mentioned, that actually we can leverage those models, right, to build on top of that. Like how do we harness all this, pre-trained those foundation models to serve a certain purpose. So I think that extra work, is much require much less computing resources than building everything from scratch.
And I just want to read it. I know earlier they said just because if you build it, they will come. So we are talking with the NSF Mayor, National AI Research Resource. It's funded by NSF. You can write a 3-page proposal and ask for cloud credits and things. But I was like, well, what's the participation like from other institutions, from people who may not have reached? So, you know, I put my money where my mouth is. So we're working on, we'll have an info session coming up to make sure that that is a resource and it's accessible wider. Also working to build a supercomputing center. When I first took this position in August of 2020, I was told that institutions in our neighborhood had us in their grant proposals for sharing computing resources. But when I knocked on the door, well, the door didn't open. So we built our own, which means we had to build it from scratch. So it's not just, I mean, it's from scratch, trying to figure out where to put it, how to sell it internally. Also looking at, we also have another project to build broadband IT infrastructure across the campuses, across the HBCUs in Atlanta, also with Tuskegee. And so it's a big effort. And it's challenging to make sure that researchers across different institution types have access to computing resources so they can participate in the research. But it's something, I'll just say it's a big challenge.
Yeah, and I just want to follow up on, you know, the Waze comment, and, you know, we heard it, you know, multiple times, you know, if they build it, you know, if I build it, will they come? You know, I think the important question that we have to start thinking about is, you know, if they build it, can I reuse it? You know, and, you know, again, with large data sets, you know, they're very hard to collect, right? So if they are refined, we want to be able to do continuous refinement and reuse that. With you know, large language models, foundation models, I mentioned, you know, how do we build on top of them so we don't have to start from scratch? Because we're now in a place where, you know, doing a lot of that initial work, we can't do it over and over again. So I think we have to start, you know, shifting sort of how we think about that to, you know, be able to do this continuous build on, you know, what we've already been doing.
Okay, yeah, thank you for all the panelists. Great discussion and summary in here. So I have one question is for the Dr. Wang and Dr. Snyder. So the question is kind of when we're trying to talk about the collaboration between the biologist and the computational scientist, because the computer scientist always trying to say, oh, give me more and more data, we can build the model in there. But for the biologist, we're thinking, show up at the models, we'll know what kind of data we need to collect. So that's kind of a gap in there, how to try to build up the best collaboration. So things we both have the biologist and computer science expert in here. I was wondering whether you can try to, from the Dr. Snyder, you can try to think about what is the essential data need collected in order to address the diabetes heterogeneity. And then is that data will be enough for the data modeling generation? Thank you.
I would argue that, you know, this is academia and what we're doing is mostly science. We don't know the answer. So therefore we collect as broadly as we can because we don't know what will feed into the models. And if you think about it on first principles, the biggest discoveries in science are the unexpected. And by definition, it's going to be that way. So, you know, most of us, you've seen already at this meeting, but it's true that we collect as much data as possible because we don't know what the contribution of every feature is into the final model. And so having said that, you do use educated judgments. You know, we assume the genome, we know the heritability for diabetes, which is okay. Well, it could be better. But therefore there is a big environmental component. And are we collecting all the things we need for that environmental component? The answer is no, it's pretty clear, right? We don't collect a lot of we haven't even talked about mental health at this meeting, and we know that impacts your glucose levels. So, you know, in a real world, you would collect everything. I guess it was touched on in a few talks, yeah, for depression and stuff, but many, many more. How about the flip side, happiness? How about the flip side of beneficial exposomes? We don't talk about that at all because people always think about the negative side. A little bit election-like, I suppose, that way. So we should be thinking about both positive and negative contributions to everything we do, I guess, and put those in the models. But it is academic science. Once you think you have pretty good, you can explain most of the variance, then you will, I guess, work on the modeling side. So long answer for the way at least I think about things.
Yeah, so what if like we want to list the 5 most important directions for the data collections? Do you think that how you're going to list the direction for the different data types?
Yeah, it depends on what the goal is. If the goal is to live a long, healthy lifespan, which I think it should be, you want to get as predictive as early as possible, ideally before birth, probably before conception. And then, yeah, what information do you need for that? Then when things go off kilter, you want to have models for how to realign them back. And that will require a different set of inputs like food, sleep, and things, I imagine. Lifestyle matters. At some point, we will be re-engineering humans. That is to say, our mitochondria go off irreversibly, right? They accumulate genetic mutation throughout life. So we will want to be swapping out our mitochondria at some point, which is actually doable. And then at what time do you do that and what are the right circumstances? That's a different, so it always comes down to exactly what the goal is. I know I broadened that quite a bit, but I actually think we will be in a world where people will live forever. So the question is, what parameters go into making all that happen?
Yeah, with that part of data, Dr. Wang, do you think that it will be enough for the AI data to do the
Well, that's, I mean, as Mike said.
A little lofty, I know.
It's science, right? So we don't know the answer. So, I mean, from my personal experience, I feel like working with the scientists is very inspiring, right? So we often start with chatting about like, what's your wish list, right? So, assume AI can do anything, like what's your wish list and what do you have now, right? And then we look at the gap and then we see, well, what we can do now, right? Maybe immediately with the current AI technology, but you can see that, maybe most part, actually AI is not quite there yet, right? Then actually inspire us to develop a new model, better method, right? That's actually help to propel our own, research in the AI machine learning field, right? So because, and then one of the breakout groups, like I was seeing that we even, talk about, how to balance this, innovation in AI model versus innovation in science, right? But to me, I think right now, I think there's a lot of opportunities that if you find the right angle, you don't need to compromise either way, right? You can, I actually I've been in the research field for more than 20 years. So some of my most cited work, right? And then that are the ones actually inspired from the collaboration. Like we start with some collaboration, originally think, okay, this may be just engineering work. We just apply this model and it works. But when we apply this model, it doesn't work as well either it's to take too long or the result is, have some kind of a bias and errors. And then we start from there to see, well, why it didn't work, right? Why we thought that would work, but somehow reality is not. Sometimes people just complain about, oh, the data is dirty, all these things, but we don't complain about it. Say, well, the data is data, right? We can't just assume that somebody going to, do all the massage of the data and then present it to us, right, for the machine learning. That can't happen, right? So basically, data is data. If this is a real bias, or whatever the issues that not this lab face, but it's going to happen across most of the people who are doing this, this kind of data collection then, or whether they can or cannot collect certain type of data, right? There's maybe certain difficulties, limits our ability to collect certain type of data, maybe too expensive, or maybe this have to be invasive technology so that we can't really do this on patient. So those kind of things. And then we'll see, and that's actually really inspire us to do things. Maybe we'll help to develop non-invasive approach, right? That would be a combination of the actual maybe biotechnology plus machine learning model, because once you collect something and the machine learning model maybe can design to better read out the signal, right? That's make it possible, right? Otherwise, it may not be possible to, if you think about something, oh, can we collect certain data or not? Oh, can we read glucose on the skin, right? So it used to be not the case, right? And then, but it's all, the difference made by the new technology, right? But then it's not just hardware, but also the software, the model on top of this. How can you really identify some very subtle signals, right? And then that's, machine learning model actually play a role in that. So I'm just thinking of that. Actually, I'd like to think, there are plenty of opportunities for people from both sides and for people already working in this, across both fields actually to really, do something together. I think, again, collaboration is the key, right? So, because we all know what, our own field, but, we really can learn from each other and do things together. Thank you. Yeah.
I have one comment, though. I do think we are there, excuse me, there is a dearth of longitudinal data. And I think for most models, I know I said it earlier, but you can't underestimate how powerful that is for both health monitoring, for understanding biological mechanisms. How do people become diabetic? Is it gradual? Is it spiked? Is it what's going on? It actually turns out it's both. We know that from longitudinal profiling. So I think we need a lot more of those kind of data.
Okay, thank you so much for the wonderful discussion. We got a question from the online audience. So in a similar way to how NIDDK-CR stores studies and data sets, would there be a public AI model repository hosted at NIH?
Yeah, I mean, that is something that we're actually sort of discussing as as part of, dkNET in the future. It's like, where can we make these, models available or information about the models available in an easy to use fashion. So again, that is something, that Wei and I and others, are thinking about. But, it is something I think that, would be good to have, so.
Thank you.
Okay, so maybe we'll end by last challenging question. I was in a chemical biology meeting before this NIH workshop, and the theme of the chemical biology meeting is data science for drug discovery. And we hear completely two opposite opinion. One part is people working on AI model. They say, oh, because of the success of AlphaFold3, we are so good to predict new drugs. And the other part is from the traditional drug discovery field. They will say, oh, after $100 million pulled on the AI model, I haven't seen even single drug enter the phase 1 clinical trial yet. So the question for the panelists is, when do you see, we know that all validation model like clinical trial, like basic science study is expensive, right? When do you think the AI model is good enough, or what do we need to do to convince people to use that model?
That is indeed a challenge. So, so myself, I haven't worked on drug discovery yet, but I recognize that's a very important field, right? So it's actually quite interesting to me that, so I think we need to not just look at the short-term benefit and gain, and also we should look at the long-term, right? So not having one new drug being, enter the phase 1, I don't think that should be considered as a failure for AI model, right? So we need to actually look into what's a, how that AI model or models being built, right? What data to be, and they already used, and what should be the additional data, additional experiment that's needed to be, running to collect the necessary data, right? Because I do believe that, even though the model hasn't said, okay, XYZ is a new drug, right? But it should already start to provide some insight, right, of the mechanism behind all those, whatever the disease that they study, and then how the chemicals and the proteins and other parts like interact with each other. This is very, very complex. And then with, for different conditions, how do we model those things? It's a very complex term. So I don't think this is a failure of AI, but rather see that opportunity for AI to really, be in the groundbreaking. Because before AlphaFold succeed, right? We never, because this protein folding field, right? They somehow hit the plateau. I mean, I shouldn't say that, but somehow the improvement has been incremental for quite a few years. If you look at the CASP, competition, right? The score do that. but the AlphaFold really, elevate that, now to the extent that you don't need to worry about, right? How do you generate that? You can just download that from AlphaFold 3, and then, you can find the structure and can use it, right? So it's completely changed the field. But before that happened, we probably, all of us doubt that would ever happen because it's so complex, right? So that's why I think, we should never underestimate the potential of AI, but also, of course, we need to do one step at a time.
Yeah, and I just wanted to sort of follow up your challenge with a challenge. Because I think the issue, when one looks at things like drug discovery, there's a lot of information that is lost, right? In terms of things that, might have been tried in, preclinical work or clinical work, and where things don't work, right? So this negative data is lost. And if we want to have better models in the future, for things like drug discovery, where it's like, does it work or not? We can't just have the data that says it works here, right? We also need the data that says it does not work there, to improve those models. So that's just a challenge.
And I'll just add, a couple decades ago, my postdoc mentor, Mike Reed at Duke, he wrote a paper. It was called "Why is Mathematical Biology So Hard?" And so we had these same kind of questions, these same sort of things back in the day when I had less gray hair. And my focus was using dynamical systems to look at cellular processing, signaling, right? And, but now we can, we have a lot more insight to protein folding. We have a lot more insight into genome. We have a lot more insight into, you can probably name a whole bunch more items that was, I think, advanced. Well, I'll say mathematics since I'm a mathematician, but okay, AI, data science, and all the rest of that through modeling. It doesn't happen in a day. It doesn't happen in a moment. It does happen over time. And I think as these technologies do improve drug discovery, that time to new drug discovery that's being, I'll say, motivated by AI, ML, and perhaps other models, I think that will shorten, and I think it will happen. I'm trying to figure out this fountain of youth he was talking about and those 2 slices of pizza, but other than that, but I think it will happen. But this is giving me flashbacks to a couple decades ago when mathematical biology was the thing, and bringing the two together. And how do we talk across the disciplines so that the biologists understand what the mathematicians say and so on. And I feel like we're having that same discussion between, well, I'll say health science researchers and those in the AI ML field. And we'll figure it out and it'll move forward.
Yeah, can I make a comment?
One very quick
Okay,
comment.
very quick. So I think I lead the AI Drug Discovery Center. AI drug discovery has been around in the industry first for almost 8 or 9 years, and they've gone through boom and bust. I don't think it's a failure of AI drug discovery. It's a failure of maybe many things, including lack of integration of chemistry with biology that we do, lack of chemistry with clinical, and also the lack of cooperation from regulatory body. And then they're stuck, like having a rocket stuck in the maybe countryside back road. And I think we need to work to expand our data set. So for example, right now, we don't have any digital chemistry data. We don't know any synthesis pathways and which one is easier. No one is talking about that. And when industry are working on optimization of proteins, and this seems to be on the other extreme of like a molecular modality, and maybe that will alienate many of us who are working at a different scale of the phenotype. So I felt that if we want to go and do drug discovery, we have to broaden our data set to put it together, and then one day we can actually, the trend is that we can do it faster and more efficient.
Thank you for that great comment. We will wrap up this session. Thank you so much to the panelists. This was a great discussion. And
we'll move to Dr. Xu Jinghua.
All right. So everything has an end. So we truly hope you all enjoyed the past 2 days. Representing the planning committee, our wonderful co-chairs, we'd like to thank everyone for your participation. Let me see if I can forward. As we all know, AI is an incredibly fast evolving field, bringing transformative potentials, opportunities, as well as many significant challenges and concerns. So obviously, one workshop cannot cover every topic, cannot conclude every discussion. Instead, we hope this workshop serve as a very good starting point for us to look into this. So this morning, we have heard from several NIDDK-funded data science programs, each working to support the community in data reuse and in leveraging AI. One of them is dkNET. So we are happy to share with you all that our dkNET team and the program staff are planning future engagement opportunities, such as virtual roundtables, forums, to keep these conversations active. So if you are interested, please consider signing up for the dkNET newsletters and following us on social media. So some QR codes are here, links are included. Additionally, we welcome volunteers to help work with us together to shape and plan future events. Last but not the least, let's hear from Dr. Cefalu, Director, Division of Diabetes Endocrinology and Metabolic Diseases, who will close the workshop.
Thank you, Zuzan. I want to thank Zuzan and Ashley and Dr. Nee for putting this together, and all the co-chairs. I think they did a wonderful group. And Zuzan said, I want you to provide some closing comments. And I said, what do you want me to do? And she goes, well, just give us your thoughts over the last day and a half. And so I looked at this and I got 8 pages that I will share with you. But I know that you have cabs to catch, so let's just forego that. But I just want to provide my impression of the overall workshop. And again, this is my 6th year at the Institute. I've seen a lot of workshops, and I actually have some metrics as to what's successful. And the first metric is just the participation and the energy. And again, we have participation here. You're still here after a day and a half, and we had participation from investigators in other countries, not only just the US. That's the first thing. Again, we had to pull people away from the mics because of the interest. So I know there was great interest. The second is just how much I've learned. And I should say how much I didn't know before I came into this particular session, and all the novel information that really was presented. And the fourth thing is, did the workshop really accomplish the objective? Did it identify the gaps? Did it identify the opportunities? And I could say that was a resounding yes. But what I got out of this is that, again, there is great potential, and for AI and ML, there's already tremendous progress made. But I think the approach is great for continuing the discovery of novel biomarkers. I know that's a great interest of us for disease progression, and particularly for diabetes. And I think, again, the approach to identifying the new subtypes that we've heard a lot about today, again, of great interest to us in DK as we move toward, hopefully, a reclassification in future years. And really elucidating the heterogeneity of the treatment response that we've heard about. I think that's going to be incredibly important. So we are just getting started. And as Zuzan said, this won't be the end of these discussions. And finally, I hope moving forward that AI and ML is going to help us address disparities in moving toward health equity. So this is a marathon, not a sprint. This is the first of, I think, many programs in this area. So I want to thank you for your participation. I want to thank you for your effort. And I want to thank you for not allowing me to be the last one in the room before you head to the airport. So thank you very much. Appreciate it.
So I want to remind everyone, we have AI workshop survey is online. Hopefully everyone, when you come back to the airport, you have time trying to finish the survey. So the link and the QR code is in here. If you have time, all the topics we discussed today is on the survey. So help to get all your input. Okay, thank you so much.